A very good morning to all of you. In the next 1 minute, we will start the webinar. Thank you. Just kindly keep your lines muted. Thank you. All right, a very good morning to all of you, and welcome to APA India 2021 Virtual Conference Webinar Series. Today is webinar number 16. The monthly theme is Emerging Challenges and Way Forward with Small Molecule Drug Development. And today's session is Regulated Bioanalysis. So once again, a warm welcome to all of you. Patanjali, can you just confirm if I'm audible, and then we can get started?
Yes, please. You're audible.
Thank you. So once again, a warm welcome to all of you. And before we start, would like to extend our special thanks to our organizers because of their efforts, untiring effort of entire year. This is such an interesting agenda we could put up on the board. So a big thank you to all of our organizers, all the presiding chair and chair-elect from different committees, including Regulated Bioanalysis, Discovery, Biotransformation, in Chemistry, Manufacturing, and Control. So big thank you to all the presiding chair and chair-elects. They were supported by a very active committee. So would like to extend our thanks to the committee members also for Regulated Bioanalysis, for Discovery Bioanalysis, for Biotransformation, and for Chemistry, Manufacturing, and Control. So a big thank you to the entire committee for their very active participation and the valuable suggestions. And the entire credit goes to them for having such an interesting agenda on board. We would also like to thank our sponsors. And many of the sponsors are with us for almost a decade now. So thank you to all our sponsors for their continuous support to AP India chapter. Shimadzu, we would like to thank Shimadzu, SciEx, Zepsci, Phenomenix, Thermo Fisher, Agilent, Syngene, USP, Natco Pharma, Vida Clinical Research, Bristol Myers Squibb, Zoetis, Nanotemper, InVitro Labs, Origin, Wuxi Aptech, and Loris Infosystem. So big thank you to all of our sponsors for their continued support. This year we have collaborated with Ministry of Skill Development and Entrepreneurship. So they have got a council called Life Sciences Sector Skill Development Council. So we would like to thank our collaborators, and through them we got access to the academia across country and to a lot of industries because Life Sciences Sector Skill Development Council, their mandate is to encourage skill development in life sciences sector. So we will be working with them in future as well. So a big thank you to our collaborator as well. Again, at the end of the webinar, as usual, we'll be doing a Kindle draw, and just would like to mention at this point that many of the winners till webinar 15, each webinar we have 2 Kindle prize winners. So many of them have already received their gift card from Amazon. And the next slot they have given is they're going to release those awards on this Sunday. So all those winners who have not received their gift, don't worry. By this Sunday, you should have your gift, surprise gift card in your mailboxes. So once again, congratulations to all the Kindle prize winners. For Q&A, all those who have joined as panelists or as attendee, please use Q&A tab. for posting your questions. After the talk is over, the moderator would take up those questions with the speaker. Suppose during the session, if we are not able to answer all your queries, then feel free to post your questions on bostondb21@gmail.com, and we will go back to the speaker and get your answers later on. So once again, if you want to participate in the poll at the end of the webinar, today at the end of the poster session, it will take place, then do attend that and you need to participate in the simple poll of few questions and you'll eligible for the Kindle draw, which we will do at the end of the poster session today at around 12:10. So finally, all the webinar talks will be uploaded on the YouTube channel of Boston International. So please do subscribe that channel. And later on, after 27th of November, all the webinars, all 18 webinars, all the talks of each of these webinars will be uploaded on the Boston International YouTube channel, and you can feel free to refer them any point in time if you wish to. So with that, I think we are ready to start our proceedings for today. So would like to invite and would like to mention that we have such a distinguished speaker today. So I'm sure all of you would be really looking forward to his talk. Russell Wiener. So before we invite him, would like to request our session chair for today, Patanjali Kadiala from Bristol Myers Squibb, to take up the proceedings further and for his inaugural address, followed by introduction of the first speaker. So over to you, Patanjali. Thank you.
Yeah, thank you, Deepak, for the kind words. Good morning, good afternoon, good evening, wherever you are, for the all APA India participants of the webinar. The monthly theme being emerging challenges and the way forward with small molecule drug development. And most of the work has been done in the low-cost countries as well as developing countries, and the outsourcing aspect has been a challenge over the years because of the patent laws or related to logistics and shipments and things like that. I just want to point out a couple of things which I was working with a CRO, which really had to, whenever the global studies were conducted at a CRO, there were several applications needed and permissions needed and permit needed for each and every study. The protocol, the IRB approval, then the individual site documents, and each study required about 10 to 15 documents, and that was a big tedious process. So we ended up influencing the CRO with whom we were working, and then asked them to approach the government of India. And then actually the head of that, the particular CRO met with Prime Minister Modi and then said, this is causing a lot of delay for all the global studies which are being outsourced to CROs based in India. So based on that multiple approaches, finally the individual study approval was waived and eliminated, so that whoever is conducting global studies analysis, there is no longer requirement for any approval. So this is like a perpetual impact, which has helped all the CROs which are working with the global trials. The second thing was the taxes, which was like 18% GST tax, levied on even the R&D type of work. So we also pursued with the same CRO, and the CRO was persistent with the Indian government. And then there again, Prime Minister Modi's government has waived those taxes, and actually the R&D's work is done without taxes now. So if any of the big pharma folks are working with the CROs, so just keep these two things in mind, which has really helped the outsourcing part of it. And now coming to Russ Wiener, I've known him for over 25 years. He's been a colleague at Bristol Myers Squibb, and he too, without any inkling of shyness, I just have to let you guys know that he was the person who kind of provided me a recommendation letter for a promotion as a senior scientist. I hope he remembers that. Coming to Russ Wiener. Russ Wiener is an esteemed biopharmaceutical professional with 25 plus years experience in global drug development, clinical trial conduct, regulated bioanalysis, and companion diagnostics development. Russ is currently head bioanalysis, biomarker development and diagnostics at Bill and Melinda Gates Medical Research Institute. Russ applies the skills gained in pharma to the nonprofit world of developing pharmaceuticals, therapeutic antibodies, and vaccines in low and middle income countries. He now focuses on TB, malaria, diarrheal diseases, and maternal, neonatal, and childrenal health which result in over 5 million deaths each year. Russ received his PhD in biochemistry from Albany Medical College and then went on to lead the regulated bioanalytical, molecular biomarker, and CDX organizations at BMS, Merck, and Daiichi Sankyo. Prior to joining the Gates Medical Research Institute, Russ' efforts were focused on oncology, specifically checkpoint blockers, and was directly involved in the development and approval of Orencia, Neurologics, Yervoy, Keytruda, and its PD-L-CDX molecule. Conducting clinical, right now the topic for today from Russ Wiener is conducting clinical trials in low and middle income countries, understanding the pre-analytic and logistics challenges. The abstract of the talk is a highly challenging aspect of conducting clinical trials is the work associated with bioassays. These challenges are magnified in low and middle income countries where one is often faced with infrastructure failures in power, weather-related aspects, availability of clean water, staffing stability, and protests. This requires constant problem solving, extreme attention to detail, always having a plan B, and most importantly, a can-do attitude, allowing one to address the needs of undeserved patients. And his passion to help patients in developing countries so that they can survive and thrive in their livelihood is a big accomplishment. I hereby invite Dr. Russell Wiener for the podium in the webinar to give the talk.
Thanks,
Welcome,
Pat.
Russ Wiener. Thanks.
Thanks, Pat. And I'll wait till I get a chance to share. If you can, the host can allow me to share the screen. So while that's going on, thank you, Pat. I literally have known Pat since he was a puppy. He's a long-term friend. I was happy to give him that recommendation. He has really, been by my side for so many years. And I can pull up the slides now. Thank you. Great. And I just want to confirm that you guys can see that. If not, I'm sure somebody will speak up. So
Yes.
thank you,
Yes.
everyone.
Yeah.
What I can tell you is Pat mentioned something in his intro, and I'm just going to read it. He mentioned logistics and shipments. And I can tell you this morning, I got a call from my lab in South Africa telling me that there was a shortage of raw materials to do some TB microbiology. And we have 3 patients that are getting ready to be dosed, and we only have enough reagents to test 2 of them. So it's an absolute reality. And it's actually, what I'm going to talk about today. And I'll go through a number of things. So I started my career in large molecules. I moved to small molecules and then genomics and other biomarker assay types. But today's talk is really something for everyone. It does not matter what you do. If you're immunoassay, LCMS, immuno-LCMS, this is something for everybody, for everyone to come together as one scientific community. And I hope when this is over, you'll really just take away a few of the things to think about. In the areas we work in, it's super, super challenging in the low and middle income areas. But as you'll see, some examples that I'll give you are right here in the Western world as well. So I'd like to start with my acknowledgements in case I run out of time. And we have Rob and from the Walter Reed, Fred from the Research Institute in Kenya. We have Eliza from the Institute in South Africa. Bob Wallace at the Aram Institute in South Africa. Carol Wallace, who is the director of the BARC Lab in South Africa. Sarika, who runs the SiteSpace Lab. And Kelly Stinson's my microbiology consultant. And really keeps me employed because she's quite amazing since I'm not a microbiologist. So I wanted to give everybody just a little bit of what the Institute is, because we're not the Bill and Melinda Gates Foundation, we're the Bill and Melinda Gates Medical Research Institute. This was formed in 2018. I was employee number 38, I believe, and I was employee only number 1 in my biomarker group for 2 years. So I was functioning, doing the same things I did as a postdoc, all the way up to what I did as an executive director when I left pharma. We're about 100 strong now, and we focus mainly on diseases that affect low and middle income countries. So we focus on TB, and to give you a feel, from 2016, 1.6 million people have died, and that stat really has maintained pretty constant. In malaria, almost 500,000 deaths, and that's likely to go up due to COVID and the inability to distribute bed netting and so on. We deal in enteric diseases right now in Shigella. Another 500,000 children have died of this disease each and every year. And recently we brought on maternal, neonatal, and child health. And this accounts for almost half the deaths that we try to prevent. And these are deaths of children who die in the first month of life, with 1 million of those from prematurity. And I'll talk about that really, a little bit, but it's the whole gamut. And we do have a rather large focus on children, both in nutrition and microbiome. But today it's all about pre-analytics. So we're going to talk about, what are our challenges. Now, I have a long history with India. I love India. Bristol Myers was heavily involved, and I spent, many a time there. I haven't been there since COVID hit. My last visit was in 2019. And you can see me on the left. I was fortunate enough to come during Holi. And it was quite an experience, but the hotel room had a worse experience. After my shower, the whole place was quite green, pretty shocking, actually. On the right are a few of my colleagues, primarily the really tall one in the center. He's the chief physician at the Gates Institute for Vaccine Research. And this was on our trip in 2019, where we were visiting hospitals, primarily the public hospitals, and visiting NICUs to see how the children were being handled and treated. And at that time, we were trying to develop a surfactant that we can deliver in a method that didn't require intubation. Because as you know, it's, intubating a sub-kilogram child is quite challenging in the best of circumstances, and in some of these hospitals were really challenging. So we went to see what we were up against, and we were not disappointed. It was super challenging. And then we went out to the regional hospitals and the smaller district hospitals to see. And this one here is literally a birthing room, a consult room, and a surgical room. It's very small, but it serves a rather large area. It's an impressive team of individuals that get an incredible amount done with very few resources. So here's the meat of the talk. What you have on the left side is a 50 mil conical tube. At the bottom is a white pellet, and that's what we're expecting. We're expecting to receive a bunch of these tubes with nice white cellular pellets, and this is what we got. And this tells us that we're not training the sites right, someone's not paying attention. And so what we did, and what we recommend doing, is we include in our lab manuals a lot of pictures. This is what it should look like. It should never even leave the building. But what I'll tell you, this was in the US. This is not a low and middle income, a place that with limited resources. This is, a high quality hospital in the US. We deal with site issues all the time. And again, this is global. We're looking at everything that's pre-analytic, everything that is sample collection from the patient, processing, usually at the site, shipment, and storage, and that affects all of our assays. I'm sure if you've been in this, longer than 5 minutes, you've had samples that have come in at the lab and they give you a call, or you're in the lab running them, and they're mislabeled. They're in a bag. They don't know if the bag is filled with one patient sample or multiple patient sample. You expect paperwork, requisition, that's not sent, or it is sent and only a few things are filled in with lots of missing information that you need to go back and query. And sometimes that information is so important that you can't even analyze the sample until that information has come back through the query system. Or you get that sample, it's the wrong patient, it's the wrong site, it's the wrong birth date, it's the wrong identifiers. So, a big, round of applause to those who are in the sample receipt parts of the labs and the CROs, because it is, it's the first line of defense against getting these samples and figuring out what they are. And everyone's favorite is when someone places a label over a label. There are issues with shipment. We expect that samples are not only put in bags and secondary containers before they're put into the shipping box, and whether that's an ambient or wet ice or dry ice, but we do expect them. But what we get is a box that is filled with samples just laying all over the ice. Or this happens all the time also. Sample didn't make it through customs. Someone didn't use a courier that was able to replenish the dry ice. And now you have samples that are toast. You know, they're no good. You can't use them. Your temp tail that's in there tells you, you know, it's been exposed at a temperature that is, you know, way beyond what you can use. If you're lucky, the temp tail will tell you that the sample actually does work. They're not cheap. They add an extra layer of data that you have to, you know, bring in and look at, you know. But I have situations where the temp tail did save the shipment. The shipment was melted. We had 5 days worth of, you know, room temp stability. It stayed actually, you know, relatively cool, just not frozen. So we were actually able to analyze those samples. So a big shout out to temp tails. And then there's everyone's favorite. It was frozen, and then it was thawed, and then they decided to put it back in the freezer. So your samples are literally in a block of ice. And if you haven't seen these situations, you just haven't been around, or like me, you're not old enough to have seen these situations. Okay, so one thing that we really do focus on is not just going to a site, and whether it's a clinical site or the lab, we want to make sure that they are doing the assay correctly. And that doesn't mean that they do the assay correctly on day one. It means they do the assay correctly throughout the study. And that study can go on for several years. So setting up a panel of QCs, basically proficiency samples that you can send to the sites, is really important. Now, a lot of people, honestly, they don't believe this. I've spoken to a number of clinical trial associates who think that you can go to the lab, you can train them once, and then life is good for the next 3 years. So here's a little bit of data. So this is a study. This was actually a study that I did at Merck, and we were evaluating PBMC preps and an assay that needed to use those PBMCs. And we trained the site, and we trained all the technicians, and they passed. So this grid would have been all green. And then right before Christmas, I got word that we're ready to go live with another study, but they never reached out to my lab to tell me that this lab was actually going live. Now, what that means in large pharma is when we potentially will not hit our final first in human at the end of the year, which will affect everybody's bonus. So not an envious place to be, but we did not want to start the study without the right controls in place. And so we said, okay, we'll start, but let us send some proficiency samples. And so on the left, you can see all the red. Green is good, red is bad. You need to pass at least 2 out of 3 to be able to move forward. Now, what's really interesting here is that 4 out of the 6 columns were technicians that had previously qualified for the previous study. Again, showing you and supporting the fact that just because you're trained once doesn't mean you remain trained 6 months out, a year out. In this case, it was a couple of years out to the next study. And so we sent those proficiency samples. We went to the site, we trained them again, and what you can see on the right is everybody basically passed. And if you want to be really a stickler, you know, technician B would be removed. But at least now we went from having, you know, 2 technicians to 5 technicians. And we recommend doing this, particularly if you're doing something that's challenging or has a reagent that could be challenging. Maybe a stability is an issue and you want to keep an eye on that, you know, every 6 months to a year in an ongoing study. So in terms of the research staff, now this is really an issue in low and middle income countries. So we sponsor a study at an institution. They run that study, and then the study's over. They don't have any additional grant money coming in. So what do they do with the staff? How do we keep the lights on in this unit while they're waiting for the next study to come in? And so what happens is these people need jobs, so they leave. And so your wonderful technician that you had in your first study may not be there. So not only do they leave, with them, they take all that institutional knowledge, all that training. It's a challenge everywhere in the world to attract and hire and retain experienced staff, particularly after COVID. There's people are changing jobs all over the place because there is such an increase in the number of studies that are happening. There's low capacity or reduced capacity when this happens. When those staff leave, you have to train and retrain them. And so you're basically down. But it wasn't just the research staff. When COVID hit, our central labs, particularly the techs and many project managers, were basically all bought away by other CROs. So it was really just one CRO kept pulling from another, and it was just a circle of people that were moving around. And we had project managers that were outstanding that were replaced with project managers that will be outstanding, but they didn't know the company, they didn't know our study, and quite frankly, it takes quite a while to get those individuals trained and ready to go. So in terms of distance, now this is one of the biggest things we are challenged with. So you can take out Google Maps and you can look at it and you could put your two cities in and you say, hey, that looks like 50 miles. Maybe it's 100 miles, right? How long could that take? An hour or two. And then you hit, run on the Google map and it tells you it takes 8 hours. And what that immediately tells you is that to get from point A to point B, it's not a paved road, it's a dirt road. Maybe it's a poor road that you can only take a bike or a moped, or some sort of small vehicle. You can't take cars, you can't take large refrigerated vehicles. And so we deal with situations where we're working with central labs, where we want to get samples, from a very long distance shipped to a central lab because we want to increase quality, robustness, fewer technicians and people to train. And it's a challenge. The time is long. Samples may not be stable. We work a lot with sputum. We work a lot with PBMCs, and a lot of these samples need to have something done to them in under 4 hours. The sputum is a little bit more forgiving depending on what type of study we're doing. But short challenges, again, you can go 10 miles and it could still take a couple of hours. When we were running studies in South Africa, there were protests, there were taxis that were being hijacked, our couriers were being stopped at these areas where they were blocking. And so, you have to have, really plan B is not an understatement. International shipments, particularly now, post-COVID, is a challenge. We have countries that want to keep the analysis in country, and we do everything we can to do that. If the assay can be set up in country, we will do that. That's actually one of our remits, is to increase capability and capacity within all the countries that we work in. There's a tremendously challenging import-export permit process, and that is not unique to dozens of countries. And just when you think you figured it out and you got your MTA in place and your export permit approved, the laws change, the government changes power, and the laws change again. And so you have to start over. It is one of the most challenging and lengthy processes, is this import-export process. Now, flight schedules to a lot of these areas were difficult to begin with, maybe you get a flight a week, and you'd have, 2, 3, 4 connecting flights. And, you've traveled, and if you've had even one connecting flight, you missed that flight, now you're stuck there. And now you got a frozen or room temp or ambient samples in boxes that are stuck in a place that may or may not be able to have access by the couriers to replenish the ice. So it's a real problem. But this has gotten so incredibly bad since COVID that where we were able to ship safety labs from point A to point B, and those flights, regardless of how many flights that were required, they would take about a day or two. And now we're seeing 96 hours and over. So what does that mean? That means we have to set up local labs and train local labs in areas that, you know, I've never worked in. Most of the people in the Institute have never worked in. And these labs are, some of them are super basic. They don't have centrifuges. They don't have minus 80s. And so we have to ship that equipment in and again, increase the capacity and the capability of those labs. And as I said, most of these things are cold chain. Logistic challenges. A lot of countries have power grid issues. In South Africa, there's an app you can follow. There's basically brownouts and blackouts on a daily basis. I've learned that when I ask a lab if they have backup power, do they have backup generators, and they say yes, I've learned to ask if they have fuel. They don't always offer that little piece of information. It can be short, it can be stolen, or it can be cost prohibitive. And of course, if you're not maintaining power, you're not maintaining humidity and temperature, which can affect your assay, especially immunoassays. And there's a tremendously high cost of updating to new technologies or even servicing existing equipment. Some areas are serviced by a single engineer, and waits can exceed 6 months to get a service call. And if you can get a service call, let's say your freezer go down, you want somebody to come in immediately. We work in areas that, you know, once it's not daylight, the security issues are a problem, and we can't let those service engineers in. Few last ones on supplies. So what we have here is areas where you can't get your common materials such as HEP blocks and IV tubing. Lots of countries don't produce their own dry ice. We ship it in the boxes that they'll ship back with us. You can't get FDA approved kits. So you have to get CE mark kits, and those performance characteristics may be different. I told you about the cold chain and the multiple shipments for samples. The same thing happens for diagnostic kits, which increases the lot-to-lot variability. And the bottom line, everything takes longer, and everything takes even longer now with COVID. So that's kind of the background and the real meat of the talk. There is so much to think about. Visit the labs, visit the sites, do trial runs with shipping samples, have backup couriers, have people in country on the ground, you know, who are familiar with customs, who are familiar with replenishing dry ice. I can go on and on and on, but I believe that probably, and I've read this in other studies, 90% of the issues that we have in the lab with poor data has nothing to do with what happens in the lab. It has everything to do with what happens before the samples get to the lab. So just to finish up here, one thing we're moving into to avoid having to ship is to send the diagnostic test to the site and do point of care testing. Just remind you again, for TB, 1.6 million die a year, 10 million new cases, and it's estimated that 25% of the world is actually has a latent form of TB, which is basically asymptomatic TB. We have sputum tests for this population. They are particularly low sensitivity for both children and those that are co-infected with HIV. We run a lot of nucleic acid tests and real old school cell culture tests, which require BL3 facilities and pretty expensive equipment, although the prices have come down, particularly in the TB area. And you know, to improve TB control, we need to be able to bring tests that are simple and accessible, you know, preferably something that doesn't require a lab. In fact, I'd prefer it didn't require a lab or water or electricity. That's really the goal we have. And so you say, why do you have to get those samples analyzed so fast? I told you about the logistic issues, but there's another issue. TB, like COVID, is spread through oral secretions and breathing and stuff like that. So what you can see in this picture in the background is a small village. Those people could just, you know, walk to the hospital. That's right there, not an issue. But this is really atypical. What we're dealing with more often, and this is Soweto in South Africa in the far background, is large, undeveloped areas. Now, this is actually, the mid-level. These are not the shanty towns. And so, yeah, they can walk, they can come over to the hospital, but it is super inconvenient. Often it's not safe. So again, this is one of the reasons, but this next picture is really the reason. So this is a sky view picture of Kilisha. And what you can see here is this goes on, if you drive down the main drag in South Africa, this goes on for a good 20 miles. These people don't have a means of travel. They're not coming to the hospital. And all you need is one person in these crowded areas to just spread and spread and spread TB. So we need these home healthcare workers to be able to go into these communities and diagnose these patients and get them on drug as soon as you can. So, really diagnosing them fast and treating them immediately is the key. This is what we have. Fast turnaround times for about 24 hours for old school staining of bacteria. The Gene Expert is an automated way of looking at the genetic material of TB, revolutionized the way we do TB research and TB treatment or TB diagnosis. On the top right is, we still do this. They still do old school Petri dish culturing. And then we can do the liquid version of that, which is fully automated. But both these tests take, a month and a half. And do you really want to wait that long to get the diagnosis? So again, let's bring the lab to the patients. This is an example of a mobile lab where everybody can come. The lab is, fully equipped. When I was in Peru, we did this. I'm gonna go back one slide. We had a van that was filled. They brought the van in, they played loud music, all the kids were excited, and they got the community to come there and participate. And the van had a portable chest x-ray, and they were able to x-ray the community, and they had some artificial intelligence type of technology. So they didn't need a pathologist, and they were able to diagnose TB in the field and then bring them to the next station where they literally gave them their drugs on site. It's quite tremendous, and it's actually a lot of fun there. They treat it like a carnival. So this is your typical point of care device. It's a lateral flow device. These are very similar to what you will see for a rapid stress test, rapid strep test, or a pregnancy test, or a rapid HIV test. They're very simple technologies, work by capillary action. You add your analyte, and whether that is blood or serum or what have you, the analyte basically gets soaked up, and there's these magnetic particles that bind the analyte. They move towards the first line, the green antibodies, where they are captured, and that gives you a red color. And then what doesn't have antigen goes to the next one, and that gives you your control line. So let's see if this works. I don't know if it'll be good to see, I'm going to pass it because it's not working over the video. You can use any sort of sample type. We have blood, everybody's favorite nasal swab. We're looking to diagnose TB with throat swabs, saliva, urine, and everyone's favorite, your stool sample. So whatever you could imagine, you can use cerebral spinal fluid, anything will go in this, but the goal is to really get things that you don't need to be invasively, don't need needles, maybe, aside from a lancet. So, this is what a rapid HIV test where they can do this, in under an hour. So it's quite remarkable. And what you're looking for is a line in the test that says it's positive, and you also need a line in the control that tells you the test is working. On the right, you can see that the control is lit up. This is supposed to be a negative test, but you can see a faint line there. So that shows you that test didn't function properly. So that's the end of my presentation. What we focus on at the Gates Medical Research Institute is really our only bottom line is saving lives. We're a not-for-profit, And we work with a number of not-for-profits all over the world, including India. So I thank you for the invite. I am super honored to come back. I believe I may have spoken at one of the first APA India meetings back in the BMS days. So again, thank you, Pat, for the invite, and thank you to the APA organizing committee for the invite as well. Thank you.
Thank you, Russ. It was a fabulous talk, much appreciated for all the background. It's pretty challenging, if any of the panelists have any questions, please come on and ask us any questions you have. Okay, go ahead, Srinivas.
Yeah, hi, Russ. It's really wonderful to see you through the virtual media, but amazing talk. I was very inspired, looking at, rather impressed by the turnaround times that you could develop for TB diagnosis. And any challenges you have seen in that space, especially for MDR and XDR TB diagnosis?
Yeah, so the what I didn't tell you is the gene expert actually does look for a couple of resistance to isoniazid and I think rifampin. So for the multi-drug resistance, I'm not sure. I haven't worked in the extreme multi-drug resistance, the XDR space, so I really can't speak to that in terms of rapid diagnosis. But usually with the people that come in with the MDR, they're going to go right on treatment, and they're going to be stopped right there, and they're going to go on for some additional testing. There are other technologies that I didn't show here from other manufacturers that can look at the susceptibility or lack of susceptibility to other key drugs. And the XDR, the really bad one, when we were in Peru, this filled up half the ward. It actually quite shocking to see that. So it actually, to me, I'm not a microbiologist. I'm actually 3 years new to this space. It's not getting any better. And with COVID, all roads lead to COVID. People aren't getting diagnosed. People aren't getting their meds. And the biggest problem we have with TB, one, getting them on treatment as soon as possible. And two, making sure people take their meds. I'm sure you know people who felt sick, they got a prescription of antibiotics, and they stopped their antibiotics halfway when they were feeling good. Well, when you do that on TB, you become drug resistant. And so what we actually do in our clinical trials is watch them. We actually physically watch every single dose that they swallow. When we go to more broader trials, we do that by video. They're called VDOTs. Visually, we observe therapy. So it is a problem. Thank you for the question.
Anybody else?
Russ, a great talk as always. I mean, it's been a pleasure to attend your talks. And just one quick question that, of course, for most of the countries which you just highlighted, I think one of the major issue is not having a point of care laboratory setup or a clinical setup out there. And as we can see that this could be the only solution or one of the major solution in such places. But when you do that, like you have shown an example of a mobile lab, so there could be many challenges with this approach as well. So would you like to just comment on that? If there are any challenges with point of care diagnostic setup having right there on the ground?
Yeah, so it doesn't, you're absolutely right. It doesn't mean that it's problem free. So I'll give you a quick example, and then I'll talk about the mobile labs. So in all of our tests, and everybody that runs drug studies understands that we run pregnancy tests. We don't want anybody going a new drug, if they're pregnant or during the study, get pregnant. And so at our sites, they have the tests that I just showed you, the dipsticks, and we have to train them how to use them. It sounds incredible, but what we learned is that people don't take the instructions seriously. says it needs 15 minutes at this stage, put it in the next one, another 15 minutes. You can get past that. But what we learned is that, a lot of these tests may be done in the early morning hours or late at night when they don't have good lighting. And then we find out that they have poor eyesight because they couldn't afford glasses. So a simple test, in your house to see if your spouse is pregnant becomes challenging. The mobile units have to get to those sites. We are looking at, really small, almost cell phone size diagnostic tests where you can do this. It's on a test strip and you put it into something that snaps onto your cell phone. We have microscopes that snap onto your cell phone. So, the ability to get those people there is one thing. The ability to get people to the point of care. I showed you a nice picture. Everybody's in a nice line. And I told you about Peru, but that took years and years of community outreach. Point of care only works if you have outstanding community outreach, because there's a tremendous amount of skepticism. There's a tremendous amount of fear of doctors in these communities. And so the only way the point of cares work is when we are actually bring someone from the community, someone who they trust, clergy, a nurse, some of the home healthcare people.
Great. I completely agree. And well said, because the only way forward I see in this case is continuous outreach program. You cannot just wait when you actually want to do your clinical trial, but throughout the year, you should have this outreach program going on. So that setup needs to be there on the ground and to be driven by the local people. Only then you can have that trust and their involvement, engagement in this large clinical trial. Thank you, Russell. Over to you.
There is a question on chat, Russ. It's Jignesh Kotecha is asking, what minimum accreditation or certification required for local labs to be qualified for analysis for regulated clinical trial?
It's a great question. There was a lab that was actually set up by the Gates Foundation for TB research, and that's right when I started, and I noticed that they did not have the accreditation from the South African, the SAFRA, which is their FDA. And I said, we're not doing work there. And boy, you thought I'd killed someone's child. It was awful. But I couldn't do it. And we require at a minimum that they have, they don't have to have CLIA or any ICH or CAP accreditation as a minimum. What they need as a minimum is the local health authority has to sign off. There has to be at least a certificate that shows that someone audited the lab. Now, that's just one step. We then go in and we do a complete GCLP audit, and we see what the issues are. We're going to hold them up to as high a standard as we can within reason, right? Because, if you're using a safety lab, they got to follow all these things. You don't want to put a patient in jeopardy. TB drug development, it's basically chemotherapy. You're giving patients 2, 3, 4 different chemotherapeutic agents, and they have nasty side effects. So we do require, now when we're for the local labs, if we're in a real desolate place, that's what we require. If we're working with a central lab, just like the EU or the US, we want to see all the big government, big regulatory agency accreditations.
Then I have a question, Russ. How about using drone technology in these remote areas? You guys are thinking about any of those aspects, or?
Absolutely. So we ran one of the early COVID studies, and we were challenged with getting drug to patients early enough in the course of disease. Just like flu, if you don't take those flu drugs in the first 24, 48 hours, they're useless. And we had long talks on drones, but they are doing it in the continent of Africa. We're not currently doing it, but we are absolutely looking at that to develop, to ship in drug, to ship in material for education and whether it's safe sex, or things, if people are in dangerous situations. I mean, there's a lot of social work that goes on primarily from the Melinda Gates side of the Bill and Melinda Gates. Bill's more on the drug development and technology side. So yeah, no, we are looking to drop into a local health provider. Here you go, literally. So we go to. I'll give you a great example. In Peru, the poorest people are in these villages that are up on the sides of mountains. You can't get up there with cars. You can barely get up there with the moped, and you got to get in there, to get these tests. So bringing up a drone and giving it to one of the local health providers is tremendous. It's not that far away.
Wow.
Hi,
Okay,
hi,
guys,
hi, Russ, it's Ravi here. So one more question from my side is related to the oncological area. So early diagnostics of any of the oncological diseases would certainly help. So in that perspective, what are your plans? Similar the case with dengue kind of a thing. So what are your plans in this area? Because it's like thirst area to work on and early diagnosis of the oncological area.
Right. So it is not one of we're primarily in infectious disease. And I didn't think there was much of that going on. I'll be honest with you. But when I visited India on that last trip, we had visited one of the hospitals, and I was surprised to see the level of interest, not in just oncology diagnosis, but in companion diagnostics, which in the US are expensive co-tests that go with it. So I'm just not versed enough in what we're doing in the World Health. Suffice it to say, at least in South Africa and in India, there's been tremendous strides and focus in that area. So I expect that what we call personalized medicine really to take off, because the technology is now there. If you can have a gene expert for TB, you can have a gene expert, particularly for liquid cancers, your AMLs and your CLLs, and the technology of genetic sequencing, it's commodity now, and you can get them at the big research centers and the CROs that are popping up. I just attended it was more infectious disease surveillance by next gen sequencing, and it's not just in Western countries anymore. It's all over the place, and it's really a matter of finding one seed university who can bring this up, right? To get the interest, to show the value, so that you can get that investment from, not only the not-for-profits, but the local government agencies. But, I see that being in 5-10 years, just like any place else.
Anybody else?
Yeah, so just one, Srinivas, before I invite Srinivas for his question, just one thing here, I think you already mentioned, but one of the important thing here is the involvement of the government, because without their support, I think this whole drive cannot be successful at all for any of the disease targets we are talking about. So especially just to share, from a TB, I think in India, there are very, very big programs going on, and I'm also a part of one of the TB subcommittee. So I just wanted to share that I'm sure you would have experienced the same thing, that in each state, there are district committees have been formed, and within these committees, there is an involvement of health workers, there are hospitals, government hospitals, and a lot of NGOs like Rotary International or such clubs who are working, and the local participation from the local areas. So that's how this whole drive is going on. And when you said just checking that each pill has been taken, I think for that, a lot of incentives are being given. And especially this NGOs plays a major role to incentivize, because continuity of the treatment is a big issue also once you are successfully diagnosed. Because if, as you said, they don't continue, then it has its own resistant issues and all. So I think a lot of effort is going on from the government also. But when we do, and when you plan any such clinical trials, unless you don't have a buy-in from the government, I think it would be difficult to really run it successfully. So your comments on that or experience on that?
Yeah, we absolutely, wherever we work, we are talking to the government. I know the Gates Foundation is talking to your government. We learned recently, I believe India has a rather ambitious goal of eliminating TB in 5 years. I mean, it's so we are there. And it's not just the clinical trials. It doesn't pay for us to do trials if the country is not going to then adopt the drug regime. Is the drug at the right price, that the WHO is willing to bring that in and support that as the new therapy? Because right now TB therapy is 4 to 6 months, and if you got MDR and XDR, you're literally 18 months on chemotherapy, where it's it's awful. I have actually some friends that got it, and it's not a happy place. And so what we're trying to do is bring that regime down to less than 4 months. We hope that trial will start actually quite soon, and we are in active talks with the Indian regulators. I think Pat had mentioned at the beginning, it's opening up a bit after COVID, and we're trying to figure out how to do those studies. But I can tell you from the top of the foundation, India is very high on the list. And if you look at where the global burden of TB is, there's a good reason. And it's not just TB, it's all the other things we're evaluating as well.
Thank you. Thank you, Russ. Over to you, Srinivas.
You're muted, Srini.
Srini, your line is muted. Yeah, we can hear you.
Yeah,
please.
Good.
Yeah, just to continue on what we are giving perspective. So India is planning to have TB eradicated by 2025, and there's a lot of thrust for even scouting the technologies being developed within the country. So I am supporting incubation center where we have a couple of startups who have AI-driven diagnosis and progression assessment tools, which government is quite interested to take it forward. And also, it's multi-pronged approach where you also look at adherence to the treatment, as Russ was emphasizing. So there's another startup which has already built a medicine reminder device, which is again AI-enabled, and so you ensure the 100% adherence to the regimen. So apart from that, I had a question, Russ, in terms of the logistics. You mentioned the challenges. So do you see DBS can play some role in easing the logistic challenges? Right blood spot or right samples?
Yeah, so Jolene White works for me, highly involved in the micro sampling space, the personalized sampling space. And we have a study in RSV that's going to be starting soon, where we are starting at the very early part of bioanalysis. We're using devices, these volumetric VAMS devices, the Mitra device, use that early in the study, use it preclinically to avoid the bridge, right? No more bridging. So we are absolutely using that for the reasons of shipping, stability. There's so many advantages. And for us, it really started not as the stability side, but we're dealing with little babies, and we need to get enough sample. And it's the newer devices are volumetric, so it's not like a blood spot that oozes and you hope you can get all that out and you did the right punch. These are pretty quantitative instruments. They look, some of them look like Q-tips and others, but no, that is right in the sweet spot of where we're going.
Yeah,
Thank you.
One last question. Sorry.
Yeah, actually, Ravi, I'm so sorry, but we are just running late. So would request you to post your questions to Boston DB21, and I promise you that I will go back to Russ and get answers for that. So Thank you, and let me invite quickly Patanjali for his concluding remarks. Over to you, Pat.
Yeah, thank you very much, Russ. This was a very fascinating and very entertaining as well as an illuminating talk. And I hope we had more time. Maybe it could have run for 2 hours or so, and with all these discussions.
I cut it down, Pat.
It was pretty fascinating. And I listened to the same talk in AAPS in San Antonio. There was a lot of interest too. So hopefully when I retire from BMS in a year or so, I'll follow you as an NGO. So thank you very much. Much appreciated. All the applause for you.
Yeah, thank you, Russ. And on behalf of the committee also,
Yes.
let me extend our invitation to you for next year. Hopefully we'll be able to do in-person meeting, and we would really have you, we would love to have you in person delivering talk here. So before we conclude, let me present you, request Patanjali to present a virtual certificate. Yeah.
This is for you, Russ, a certification of appreciation presented to Russ Weiner for his talk on conducting clinical trials in low-income countries, understanding the pre-analytic challenges. This is our recognition for you, small token of appreciation, Russ. Hopefully we'll meet you in person next time, and then we'll go from there. Thanks
Sure.
a lot.
Thank you, Russ. Thanks a lot.
Send my regards to Adam.
Sure.
Sure. Thank you.
Take care, guys.
Okay. Thank you, Russ. Thanks, Patanjali. And would like to thank all the participants, attendees, and panelists also for their active participation. All right, so I think we are ready to start with our poster session. So let me just display here. Patanjali, are you able to see my screen?
Yeah.
Okay, great. So would like to welcome, after very, very interesting talk by Russ, now we are ready to start APA India poster session. So just would like to invite all the poster presenting authors. A warm welcome to all of you. And just for the audience, would like to inform or share that in total there were 45 posters, a poster abstract that was submitted, and they were duly screened for their relevance and the content and novelty and those things by the panel. And finally, we have shortlisted 15 posters for the final APA India 2021 poster session to be presented today. So today you'll have 15 posters. So each poster presenter will have around 6 minutes for his talk and 2 minutes for Q&A. So like that, in total, 8 minutes of slot is there for each poster presenting author. And within that 2 minutes, the judges, the panel of judges, they may ask you any questions. And then we'll proceed to the next poster presenting author. So this is the flow, and each one are given a particular slot. I know we are running a few minutes late, but we'll try to cover up later, no worries. Each presenting author will still get 6 minutes for their talk and 2 minutes for Q&A. So after 6 minutes, maybe I will just give a buzz so that you know that your time is over and you need to stop the presentation. So with that, I think without further ado, let me first, in the first poster is going to be from Ishwara Raju Kosori. The poster title is Analytical Methods for Determination of Nitrosamine Impurities in Selected Sartans and Ranitidine Drug Substance. So while I invite him, very quickly, let me just check about the judges as well. So I'll just stop sharing, and just in a minute we'll start, and let me just see if all the judges are present. A quick roll call, and would request all of them, I mean, it's up to you if you want to switch on your video, but just want to make a roll call. So we have Amarnath. Amarnath,
yes, Deepak. Yes, I'm here.
Okay, great. So Amarnath is there. Let me just very quickly add spotlight for him. We have Shefali right here. We have Ravi already present with us, and Shrikant, are you there? Yes, he is very much there. And Hemant, I think maybe he would be joining. Sam, Samula is also there, so maybe you can switch on your video later. And Jignesh, Okay, so Jignesh was there, so hopefully he'll be joining soon. So, okay, Jignesh.
Yeah, I'm here, Deepak.
Great. So I just wanted to highlight all these people that I have nothing to do with the posters. These are the judges. So later on, if you have any complaints, these are the people to reach out to. Anyway, so jokes apart, but I'm so glad. Just would like for the poster presenters, would like to say that we have got a distinguished panel of judges: Patanjali Kadiala from Bristol Myers Squibb, Amarnath Jaiswal from Viatris, Dr. Shefali Gupta from Clienta, Jignesh Kotecha from Torrent, Shrikant from Laksai, and Ravi Shankar from Sun Pharma. So all of them are distinguished, have been in the field of drug discovery and development for almost more than 20 years now, and with loads of experience with them. So they are going to be judges, and they will be asking few questions based on your presentation. And then at the end of it, they'll be scoring based on what they felt about your presenting poster and the content, the oral presentation, visual slides, the novelty and overall impact of your presentation. Okay, and for our judges, I hope all of you have that Excel sheet which I've shared for the score sheet. Can we have quickly confirmation, Ravi?
Yeah, Deepak.
Okay, Jignesh, Shrikant, Dr. Shefali, and Amarnath, do you have the Excel sheet?
Yes,
okay,
we have.
All right, so that's it. So no further rules for that, and I think we are ready to start. So let me first invite Ishwara. Okay, Ishwara, feel free to share your screen. I will make you the co-host. I have already made you the co-host, and let me first.
Screen is visible.
It's very much visible. So over to you, Ishwara. We can see you, and your time starts now. Please proceed. Thank you.
Yes. Yeah.
We can see your slide, Ishwara. You're right there. I mean.
Yeah, I'm having something black in my screen.
Oh, okay. No worries. I mean, take your time, maybe.
Yeah. Good morning, everyone. This is Ishwar Raju, working as a senior scientist in USP India Private Limited. Today, I'm here to present our poster. Topic is analytical methods for the determination of nitrosamine impurities in selected sartans. Selected sartans here are 5 sartans, that is valsartan, volmisartan, irbesartan, telmisartan, and losartan potassium, and ranitidine drug substance. Here authors are including me, our USP India colleagues, as well as USP US Rockville colleagues. As you all aware, nitrosamines have become a major concern for pharmaceutical industry and health authorities due to its highly toxic nature and detected in multiple drug products. Actually, it was identified in the year of 2018, and that time NDMA, initially NDMA was discovered in valsartan drug product. Because of this, regulatory agencies immediately alerted the manufacturers and recalled many drug products from the market. After thorough investigation, regulatory authorities finally concluded that it is due to the formation of these nitrosamines, dimethylamine. It is in the solvent dimethyl format together with sodium nitrate in the presence of acid may lead to the formation of this nitrosamine in the manufacturing of certain drug substances. Additionally, they also mentioned that the contamination of solvents, reagents, excipients, and packing materials will also lead to the formation of these nitrosamines in the final drug product. As a result, health authorities provided a guidance diligence and risk assessment, risk evaluation, and appropriate analytical testing of nitrosamines in the products. In order to support the manufacturer as a standard setting organization, USP extensively worked on this issue, and finally, we have come up with a general chapter, that is 1469, where we have proposed 4 different analytical methods. So 2 methods are related to GCMS and 2 methods are LCMS. These detection limits are ranging from 5 ppb to 50 ppb. Additionally, in-house we have developed one more procedure for NDMA detection in ranitidine, which was not published in the general chapter. Actually, introduction nitrosamines is a general term used in designate a vast group of N-nitroso compounds. Why it is important? Because these impurities are very toxic in nature and mutagenic and carcinogenic properties. This induced a cancer. Due to this reason, these impurities are highlighted and mainly as per concern where the N-nitroso group is present. Mainly, this poster provides information on USP's efforts to address the nitrosamine concerns in drug supply. As an immediate approach for patient safety, published a general chapter, provide a science and risk-based approach for the control of nitrosamine impurities to ensure the potential presence of nitrosamines in drug substance and drug products is identified, assessed, and controlled. We have proposed 4 analytical methods as mentioned earlier. 2 methods based on GCMS. GCMS, we know 2 sample introduction techniques are there. One is headspace and one is direct injection. We have 2 independent methods are proposed. Each method is having their individual importance. Headspace, we know the advantage is we can eliminate maximum sample matrix interference. That's why we have proposed headspace method also in the general chapter. These are the left side, you can see the chromatographic conditions, and right side you can see the mass spectrometry procedure details. LCMS also, we have proposed 2 procedures for Saturns. Procedure 1 is based on a high resolution mass spectrometry, where we have used Orbitrap equipment, and another procedure is a triple quadrupole LCMS technique. We have used it. Here also you can see the chromatographic techniques in the left side, and right side you can see the mass spectrometry parameters. Apart from this, NDMA detection in ranitidine, NDMA was identified in many drug products of the ranitidine by FDA and other regulatory agencies. This procedure also we have validated in-house. The suitability of the proposed 4 methods was assessed in-house and validated. The result shows that all are meeting the validation requirements. Specificity, there is no interference observed in the proposed procedures due to the unknowns or other matrix interferences. And linearity also, we have seen a good linearity against to the concentration. And accuracy, we have seen the good accuracy result, plus or minus 20%. We are well within the criteria. And intermediate precision, we have tested with other brand of equipments as well as other lot of columns with a different analyst. We have tried the proposed methods with multiple vendors in the current trends. Methods are working well. And if you see the right side, the results of 4 procedures are summarized here. Mainly HRMS method, we can determine 7 nitrosamines at a time for 3 Saturns, valsartan, irbesartan, and losartan. Here the sample concentration is 20 mg per ml, and 5 microgram is our LOQ. And procedure 3 is for
Ishwara, yeah, Ishwara, 6 minutes is over, so maybe you can conclude in next 1 minute quickly. Thank you.
Sure. Yeah. Here, each method is having their individual importance, and here maximum methods we have used an internal standard to get a good reproducible procedures. We have varied the concentration based on the technique from 20 mg per ml to 100 mg per ml, and our LVQs is 5 ppb to 50 ppb. And these are the representative chromatograms. Nitrosamines are well separated each other, and these are separated from the matrix interference. Mainly, we have proposed chromatography coupled with mass spectrometry methods. These procedures have been established to support the manufacturers for meeting the regulatory requirements and patient safety. Multiple analytical procedures provide a best choice for the pharmaceutical industries to produce a high quality drug substance and drug products by controlling a potential nitrogen impurities. These are very helpful method, and currently chapter is in official, and many manufacturers around the world they are using this method, and we are very much happy. These procedures are giving some guidance to the manufacturers to control the nitrosamines and to meet the regulatory requirements. In turns, it is very much helping to the patient community. These are the references, mainly USP general chapter and FDA guidances and FDA testing procedures and ICH guidances. With this, I'll close my talk. Thank you all the participants and the organizing committee and the presenters. I'm happy to take the.
Thank you. Over to the judges. Feel free to ask any questions you may have.
Ishwara, this is Amarnath. I have just a quick question.
Yeah.
Since we have a developer to, to quantify the NDMA with the different techniques, right? Have you challenged those method by taking any of the commercial product? And if you have done it, did you find any differences using the various technique in terms of quantification of NDMA?
NDMA, what we have done, FDA method, we have taken as a reference. With that method and GC method also we have verified, and GC method we are seeing erratic results. That's why FDA method is giving a good reproducible results, and we have modified the conditions as per our internal equipment response sensitivity. And
my question is, let's say you have the method with the GCMS as well as the LCMS, right?
Yes.
When
yes,
you
yes.
have taken a product and when you analyze those product in terms of quantifying the NDMA using the GCMS as well as the LCMS, did you find any differences in both the techniques?
Yes, GCMS, yeah. NDMA granted in some issues there. I think FDA also highlighted in their documents. GCMS is not suitable since NDMA is generating through the molecule itself in the GCMS conditions. That's why LCMS is preferable for a ranitidine. But we got a reproducible result in the same technique, not verified with, because issue is there with the GCMS.
Ishwara, a quick question from my side, Ravi here. So what is your contribution here? I mean to say, we know that USP is working together, a lot of methods on that. So what is which method you have contributed here for a development? Sorry, I could not hear.
Yeah, so his question was, what is your contribution in this work? Because USP is anyway doing great science. So in this entire work, what was particularly your contribution? I think, Ravi, is that right? That's your question?
That's correct.
Yeah, so Ishwara, over to you.
I think he's muted, I think, or maybe it's my.
Yeah, Ishwara, you have muted.
Yeah, Ishwara, we can't hear. Yeah, you have to unmute yourself.
Now it is audible?
Yeah, yes. Audible. Please proceed. Yes.
Yeah. My contribution to this procedure is we have evaluated these procedures in a different manufacturer equipments, and we have validated the procedure.
Okay.
Did I answer your question?
Yeah. So, Ravi, did you get the answer?
Yeah. Thanks, Ishwar. Yeah.
All right. So, thank you, Ishwara, for being the first poster presenter, and good luck to you. And thank you for presentation. You can now stop sharing.
Yeah. Thank you.
All right. So now we move on to our next presenter, Ashish Kumar Sonkar. Ashish, if you're there, let me just quickly check because I did not see his name out here. Okay, so I couldn't see Ashish. Maybe he was there initially. Okay, no worries. So we'll move on to the next speaker. So for the judges, would like to say that the poster number 2, Ashish Kumar Sonkar, he is not there right now. So so now we are moving on to poster 3. So poster 3 is from Zifang Yu from Wuxi AppTech. So they are one of our sponsors, and it's a sponsor presentation. So it's not for the. I mean, you don't have to judge that, but they will get to present their work in 6 minutes time, and in another couple of minutes, we may have any questions you can ask. So let me invite Zifan.
Hello.
Yes, Zifan, we can hear you. And
Yeah,
now
but I cannot share my screen. You have to give me, yeah.
Yes, you should be able to share now. Yeah.
Okay, great. I can do it now. So can you see my slides?
Yes. And if you can switch on your video. Yes, we can see you. So I will just.
All right. Well, this is great. Well, good morning, guys. Well, this is Jifeng Yu from Boston, United States. And I would like to thank APA for inviting me to present my poster, Wuxi AppTech Hits. Well, I cannot see something. It's blocked by, let me see how to do this. Let's see.
We can see your pointer. I mean, if pointer is what you're looking for.
Yeah, because my title is actually covered, you know, by those things. Yeah. How do you do that? Let me see.
Because we can see your complete display.
Let me see.
Maybe go out of presentation mode and see whether you can see that, and then you can come back to presentation mode.
Okay. Well, anyway, anyway, it's fine. Well, it's my great pleasure to present my poster here, particularly for friends in India. Okay, so first I want to briefly introduce Wuxi AppTech. Wuxi AppTech is a CRO company dedicated to contributing to the global healthcare community. So who am I? And I'm a director working in Wuxi AppTech Hits, which is a unit under Wuxi Biology. So today I want to introduce you, the major technology in Wuxi AppTech Hits, the DNA encoded library. Using this technique, Wuxi AppTech Hits actually provided services to customers worldwide. We can offer a service covering the whole target to hit platform. We can purify proteins for our customers. We run DEL selection and fragment-based screening for our customers. And the compounds identified from those screening can be either synthesized by Wuxi AppTech or our clients themselves. And we have developed biophysical assays and biochemical assays to validate those compounds identified from the screening. And in the end, we can do co-crystals. We can provide co-crystal services to enable structure-guided drug discovery and lead optimization. And Wuxi AppTech Hits has 4 operational sites located in China, United States, and Germany, and with more than 280 proficient scientists, including biologists, chemists, and cheminformaticians. We are very confident to deliver high-quality data and to meet all the deadlines. So within the last 5 years, Wuxi AppTech has established its DEL platform in which more than 150 libraries containing more than 60 billion drug-like compounds have been created. And this is a DEL compound, which is constructed by the attachment of chemical building blocks to a molecular scaffold, followed by the ligation of its corresponding barcode. So a DEL compound is actually a chemical structure covalently linked to a DNA barcode. So by using those DNA barcodes, billions of these DEL compounds can be poured together in a small Eppendorf tube and incubated with purified recombinant proteins for affinity selection, and enriched affinity binders can be further identified by next generation sequencing of those DNA barcodes. And because of the similarities between DEL molecules and PROTACs, DNA encoded libraries have been considered as optimum starting point for finding protein degraders. Well, to better serve our customers with different needs, WuXi AppTec actually is providing different, very flexible business modes for our customers, including DEL Pro, DEL Lite, and DEL Open. For DEL Pro, customers need to come to us with their target protein identity. So we will do FTO check to make sure there are no conflicts. And those targets will be screened against our largest library pool with more than 46 billion drug-like compounds. And more importantly, more than 6,000 homemade novel scaffolds or building blocks have been incorporated in those libraries, which significantly increased the diversity and novelty of those libraries. So all the experiments and analysis will be performed by WuXi professionals, and in the end, selection data and chemical structures recommended for those binders will be provided to our customers. Well, next is DEL Lite. Well, DEL Lite is designed for customers who cannot release or disclose their target information. And for those customers, they can directly buy our DEL libraries containing more than 15 billion drug-like compounds. So the DEL selection will be performed either in those customers' laboratory, or the customer can come to one of our teaching labs where professional guidance can be provided by WuXi AppTec with a small charge. So the customers will have to send back those samples to WuXi AppTec for further processing, sequencing, and analysis. And WuXi AppTec will present selection data, including enrichment score and copy counts of those binders. And at that time, customers can decide whether they want to pay another fee to release all the chemical structures. Well, the last business mode is DEL Open. Well, DEL Open is actually exclusively designed for investigators in academia. Well, for investigators in academia, they can request up to 4 billion drug-like compounds, and they can do selections by themselves. And also they can keep their target information. They don't need to disclose this target information initially. So when they finish their DEL selection, they need to also send back their samples to WuXi AppTec for further processing and sequencing. Still, WuXi AppTec presented, the selection data, and investigators can, choose up to 5 compounds to be synthesized by WuXi AppTec, at a discounted charge. So investigators can, test those compounds in their own laboratories. And once some compounds have been validated, investigators can come to us and exchange their target ID, for chemical structure information from WuXi AppTec. So in my last slide, I would like to briefly introduce our newly launched.
So Zifeng,
yes.
Sorry to interrupt, but 6 minutes is over. So maybe in 1 minute you can conclude. Thank you.
Okay, great. This is my last slide. So I just want to briefly introduce our ready-to-go assays. Well, to validate our compounds identified from our screening, WuXi AppTec has, developed, quite a number of biophysical assays and biochemical assays. For example, this nano DSA, servo shift assay, MST, SPR, ADP glow assay, and nucleotide change assay, nucleotide exchange assay. So for targets listed in this table, biophysical assays and some biochemical assays have already been established. And if customers are interested, they just need to bring their compounds to us, and we can do flexible assays, either single assay or multi-assay in parallel for our customers. And those assays, they are cost effective. We can deliver high quality data, and the turnaround time is pretty fast, up to 2 weeks. Well, thank you very much. I want to stop here, and I'm happy to take questions.
Thank you, Zifeng, and would request you if you can stop sharing so we can highlight your video. Yeah, thank you.
Sure.
So it's an open forum for a panelist or attendees. If you have any questions, feel free to ask him. So I have one question for you, Zifeng. Great presentation, and I think it's a real interesting tool. But already you have mentioned about confidentiality, and I see where the application is. I think everything revolves around the target selection itself. So did you really face any challenges when you go out with this product to the potential companies from a confidentiality point of view? Do they feel hesitant or any issues there to sell this product to them, or because I think everything revolves around the structure, the basic structure, so just wanted to check with you that how do you address this?
Okay, this is a great question. First I want to mention, WuXi AppTec doesn't own any IP. This is first. Second, well, for DelPro, the only target information we can know is actually from DelPro initially. So when we receive customers target information, we will do FTO check to make sure there are no conflicts. Okay. So and another for Del Light, we don't know their target information. So we don't know that. And for Del Open, that's interesting because that's for initially, we still don't know their target information. But when we finish the selection and when we have a meeting with academic PIs, well, they can request, say, well, they want to synthesize the best 5 compounds, and then they will do assays in their own lab. Okay. And if they found, well, the compound is working, and they will come to us and they will say, well, I can tell you my target information, and now you tell me the chemical structures. And then we will remove those 5 chemicals from our library. And in the future, even, another PI comes to us, with the same target, and those 5 compounds will never be disclosed.
Okay.
Yeah.
Right.
Yeah.
No, thank you. Thanks a lot. Any other questions from panelists? Jignesh, Ravi, Amarnath, Dr. Shefali, or any other attendees? Yeah. Okay, so in that case, would like to thank you, Zifeng, for your time, and I know it's late for you, so thank you for taking this presentation late in the evening, and it was really interesting, and looking forward to another talk from WuXi next week, which is we're going to have a detailed discussion again. So thank you, Zifeng, for a wonderful presentation. Thank you.
Thank you. Bye.
Thank you. Bye-bye. So let's move on to the next. Now we have Rahul Chandonkar. He's going to talk about development of stability indicating assay method for determination of pimozide in the bulk drug. So for the judges, it is poster number 4, and the presenting author is Rahul. So let me invite Rahul, if you can share your screen, and then you can get started.
Yes, sir. Good morning to all. My name is Rahul Chandonkar. I'm assistant professor at Goa College of Pharmacy. The title of the presentation is Development of Stability Indicating Assay Method for Determination of Pimozide in the Bulk Drug. And I have done this work under the guidance of Dr. Anand Mahajan, who is Associate Professor, Department of Pharmaceutical Analysis, Goa College of Pharmacy. A brief introduction about the drug. So pimozide is a long-acting neuroleptic drug which is used in the management of schizophrenia and also to relieve the symptoms, that is vocal tics, which is associated with Tourette syndrome. So pimozide has got a molecular weight of 461 and a pKa value of 7.3. Structure of the drug is as seen in the slide. We did an extensive literature survey to pull up all the available methods which were available for pimozide. So we came across a few analytical methods for determination of pimozide in formulation as well as plasma. However, none of the sources which we came across had carried out extensive stability testing of the drug under the ICH mandated stress condition. So the methodology that was used was first development of a HPLC method in order to monitor the drug degradation behavior, followed by post-degradation studies, then optimization of the HPLC method, for example, to improve the resolution of the impurities, then validation of the analytical method, and finally perform LC-MS studies. So the first, initially, to monitor the drug degradation behavior, a simple isocratic method was developed. Was done on a phenomenic C18 column having dimension of 150 into 4.6 millimeters and particle size of 5 micrometers. Mobile phase used was ammonium acetate and acetonitrile having ratio of 60:40 volume by volume. pH of the buffer was adjusted to 3.7 with glacial acetic acid. Detector was set to record absorbance at 280 nanometers as that was found to be the lambda max for the drug. Flow rate was 1 ml per minute and the injection volume was 10 microliters. This is the chromatogram that we obtained after following the conditions which I have just now said, and the retention time was 7.5 minutes. So stability studies was carried out under the recommendation of the ICH guideline, ICH Q1A R2 guideline. So the drug was subjected to hydrolytic degradation under acidic, basic, and neutral condition. Further, it was also subjected to oxidative, photolytic, and thermal stress condition. So the stress studies were carried out both at room temperature as well as elevated temperature at 70 degrees Celsius for 7 hours. After performing the stress studies, it was clear to us that drug is extremely resilient to hydrolytic degradation in presence of acid, base, or neutral condition. Also, it was stable to photolytic and thermal degradation. However, it was observed that the drug degrades in presence of 15% hydrogen peroxide at room temperature for 2 days. So this was the chromatogram that we had obtained, of the pimozide and its oxidative degradation products in the isocratic method that we had developed initially. We see that impurities are eluting very close to the void volume, and also the resolution in between the impurities is not very good. So we had to optimize the method. So we came up with a gradient method, and in this gradient method, the time and the composition parameters is as seen here. So we got very good separation from the void volume, and the resolution in between the impurities and between the drug and the impurities was very good. We also found that this method was very selective, and the selectivity was established. It was specific and selective. The specificity was established by comparing the chromatogram of the degraded sample with a blank which was stressed at a similar condition, and the selectivity of the method was established by determining the peak purity value, which was found to be about 99%. Further, the drug was subjected to analytical validation as per the ICH Q2 guidelines. So the method was found to be linear in concentration range of 20 to 100, and the correlation coefficient was found to be 0.999. Intraday and interday precision studies was carried out at 3 concentration levels, 30, 50, and 100. There was no significant change in the peak areas. Also, the percent RST value was found to be less than 2%. Accuracy study was carried out by the means of standard addition method. at 3 concentration levels, 40, 50, and 60, that is 80, 100, and 120% of the target concentration being 50 micrograms per ml. The recovery was found to be very good, close to 100%. We used this method to study the LCMS-MS fragmentation pattern of the drug. So we got the spectra, LCMS-MS spectra is as seen in this slide. We got a few peaks, 8, having MBI 203.9, 108.7, 217.1, and 201.3, and so on. So with this data, we were able to construct a nice fragmentation pathway. So we can see that drug follows 2 distinct fragmentation pathways highlighted by the red and the black arrows. So looking at the first fragmentation pathway, we see that the drug loses this benzimidazole moiety in order to yield a fragment having M by Z of 320.
Rahul, sorry to interrupt you. You have now, I think time is up. So 30 seconds, you can conclude. Thank you.
Yeah, yes, sir. 328.19. You know, we were unable to, find out the LCMS of the impurities and the structure. We could not determine because that work is still in progress. So we are planning to perform LCMS and LCMS-MS studies on the impurities in order to determine its structures. Yeah, that's it. These are the references. Thank you.
And now request Rahul, if you can stop presentation, then we can highlight your video. Yeah. I mean, you have to say stop share.
Yes, sir.
Okay. So
quick question. When we are
sorry, I could not complete the presentation properly.
No. It was completely fine. No worries. You were on last slide anyway. So yeah, over to you, Ravi, please.
Yeah, Rahul, when we see the chromatogram, there were a lot of baselines. So are you happy with the development of a gradient method? Or so?
Yes, sir. So the thing is that we have only one system at our college. We understand that there was a lot of noise. That was because of the system, the detector had some issues. So we are academic institute. So we have certain limitations with our instrument. This is not only for this particular method, but the other drugs also. There is slight disturbance in the baseline because of some detector issue with the instrument.
Okay, understood. Yeah.
Rahul, this is Ignesh. So you referred that
Yes,
you got the recovery by standard addition method close to 100%,
Yes.
but
Yes.
what we could find on slide number 9 in the range of 40 to 60%. You can correct me if it is
Sir,
so.
that yeah. So the target concentration was 50 micrograms per ml. Now, so it's 80% is going to be 40 micrograms per ml, and 120% is going to be 60 micrograms per ml. So it's not in percent, sir. It is in concentration: 40 micrograms per ml, 50 micrograms per ml, and 60 micrograms per ml.
Okay. So it was presented in terms of what you got in terms of microgram per ml.
Concentration.
Concentration.
Yes,
Okay, fine.
Yeah.
Thanks.
Rahul, this is Srikanth. I have one question. So what LCMS you used for the checking of the impurities?
So we use CIMARDU instrument with the quadrupole detector.
Okay.
Triple quad.
So whether the same method was adapted for the HPLC, whatever the method?
Yes, sir. Yes. The same method that we developed was adopted for the LCMS-MS studies.
Okay. Thank you.
And Rahul, another question, have you analyzed any of the marketed samples for this degradation, peroxide degradation?
No, sir, we have not analyzed any marketed formulation because we were more interested in finding the structure of the degradation products of the pimozide. So as of now, we have not tried any marketed formulations, but I have got some suggestions from my colleagues to also try this method on formulations that are available. So I'm planning to do that, sir.
Okay, thank you.
Yeah, no more question, Deepak. We can.
Thank you so much. Thank you so much for listening to my presentation.
Deepak, are you there?
I think we lost him. I think.
Yeah.
Let me WhatsApp him. I have the WhatsApp. So just let's see.
Yeah, can you hear me, Patanjali?
Yeah.
Yes. Yes.
Okay, sorry, I'm so sorry, I was muted. Okay, so doing a lot of mute unmute, so I got muted myself. So sorry about that. So we are now going on to our next presenting author, Ashutosh Shaler. We all know him from Shimadzu and have heard him earlier also. So Shimadzu is our sponsor for over a decade now, and Ashutosh is going to talk about charge variant analysis of MAP biotherapeutics by Nexera XS UHPLC with a Shimpack Bio IEX column. So for the judges, you don't have to judge this particular presentation. So let me invite quickly Ashutosh. Ashutosh is there. And yes, Ashutosh, you should be able to share your screen now.
Thank you, Deepak, for the nice introduction. I will just share my screen.
And also, Ashutosh, if you can switch on your video.
Yeah. Hope my screen is visible. Can you quickly confirm, Deepak?
Yeah, yes, Asutosh.
Hello?
Yeah, we are able to see you.
Yes, we are able to see you. Only your video is off. If you can switch on the video.
I have already switched on my video.
He's on.
Okay, great. Please proceed. Yeah.
So again, very good morning to all of you. Today I will be presenting the charge variant analysis of monoclonal antibodies using UHPLC from Shimadzu and the specific columns, the ion exchange columns from the Shimadzu. So these are the authors, and we have conducted this study at our lab located at Mumbai. So to begin, the initial part basically.
Ashutosh, sorry to interrupt. I think there is some background noise coming from your side, and I can see the bandwidth, the network bandwidth, it is showing low for you. So maybe you can switch off your video. It should be okay, but maybe it will improve. Please proceed.
Yeah, please whether my voice quality is.
I'm hearing some background. Can you just confirm any of other panelists? You are also hearing some background from.
Yes, there's some static. Yeah.
Okay, so Ashutosh, maybe if you can remove the headset and try, maybe that may help, because or you can check that connection, because some static background is coming from your side. Yeah.
Yeah, I don't know, something in the background, some instrument is working, then probably we can hear that kind of.
No. I'm at home.
No instrument. I think Ashutosh, if you can just remove the headset, because sometimes that connection creates such a sound. So if you can remove the headset and use the laptop speaker instead, the phone mic.
Yeah, just disconnect. Can you confirm?
Say a few words.
Hello. Yeah, am I audible now?
You are audible. Okay. Anyway, I think let's continue, because in interest of time, there is some background, but you can please continue. No worries. We can hear you clear. Thank you.
So to begin with the introduction part, the monoclonal antibodies, these are the recent or modern medications, I can say, to fight certain dreadful diseases. For example, the cancer, the autoimmune disorders, several infectious viral and bacterial infections. It has several advantages. One of the important advantage it offers is the high specificity. It targets the specific antigen and it kills that particular antigen in the layman language. However, having said the advantages, there are several challenges. Because the monoclonal antibodies, these are the protein molecules having a very huge and complex structure, the characterization and the identification becomes a challenge, a task. Slight changes in during the manufacturing process leads to the heterogeneity onto the monoclonal antibody. The heterogeneity leads to the adverse events. So the regulatory bodies, they implement several physical, chemical, and biological tests known as the critical quality attributes to keep a close eye on to monitor the heterogeneity amongst the different batches or the lots of the monoclonal antibodies and to ensure the quality. The charge variants are several heterogeneity which occurs amongst the monoclonal antibodies. When I say the heterogeneity, it will be in terms, it can be in terms of the size, the hydrophobicity, and the charge. In this we'll be speaking about charge variants. There are techniques, analytical techniques used to identify the charge variants of the monoclonal antibodies, and it should be minimum during the manufacturing. Once after manufacturing, when the monoclonal antibody comes into the final stage of the purification and formulation, the charge variance has to be minimized. So the techniques which I was referring is electrophoresis and the cation exchange chromatographic technique. Electrophoresis technique, it has certain limitations like the reproducibility, the operation part, as well as accurate quantitation. These limitations have been overcome by the chromatographic technique, the cation exchange chromatography. Again, in the cation exchange chromatography, there are two types of modes of solution. The salt gradation uses these high concentration buffers gradient to remove the charge variants one after the another from the column, and the another method is pH gradient. So we have used the salt gradient method, which is a conventional method and which has been mentioned in the regulatory guidelines. So we have performed this study using a UHPLC from a Shimadzu known as N series, with a dedicated column, I can say, for the charge variant analysis, which is known as the Shimpact Bio IX columns. This particular system, I can say, is a biocompatible system which can tolerate high concentration of buffers, which is used especially for charge variant analysis with very low carrier effect because of a specific design of the needle and the injector port, as well as there are several analytical intelligence features which are offered by this system, wherein you can remotely monitor the status or the acquisition. You can remotely perform all the operations. You do not need to be present in front of the instrument, which is must during the new normal of this analytical new normal life after the corona pandemic. So this ease of operation is improved in this particular system. The chromatographs for basically we perform this study for onto the four monoclonal antibodies, namely the trastuzumab, the omalizumab, and the rituximab and rituximab. So you can see a good separation, a good resolution in between the main component. This is the main component followed by the variants. The acidic variants, which elutes first because of the cation exchange, and the basic ones, it elutes later on. Similarly, for omalizumab, cetuximab, and rituximab, you can see a good resolution between the main component and the charge variant. This is the statistics, analysis statistics. We have performed this study for 4 maps. The relative area percent is calculated for the main component, followed by the acidic variants and the basic variants. You can see for cetuximab, there are more charge variants, I can say, because the percent of the main component is 42%, and the charge variants occupies the rest of the acidic and the basic charge variants occupies the rest of the composition. The trastuzumab, we have procured trastuzumab from the market, whereas the rest of the monoclonal antibodies were from the customers, provided by the from the customers or by the customers, followed by the repeatability test. Of course, this is very important because using the high concentration of the buffers over a period of time, maybe more than 4 to 5 days. We it was very consistent against and the percent repeatability, the percent RSD when the samples were injected, same concentration was injected throughout different days, inter-day. The percent RSD found for almost 20 injection, it is less for all the charge variants as well as the main component for all the 4 different monoclonal antibodies were less than 5%, which proves the ruggedness of the method as well as the robustness of the instrument, which is, as I said, the Nexera UHPLC from Shimadzu is a biocompatible UHPLC. This is all about my talk regarding the charge variant analysis. Now I'm open to take the questions.
Thank you, Ashutosh. As always, very new technologies, and thank you. So, any questions for you can stop sharing, Ashutosh. Any further
I
questions?
already stopped.
Okay, great. So, any questions for Ashutosh from any of the committee members?
Yeah, Ashutosh, Dr. Ravi here. So, so whatever the method you have shown, it's one method, or like varying a gradient, you could achieve this, because generally each maps behave differently when in terms of charge variant, and there may be a lot of hidden peaks when we consider acidic variants as well as basic variants. So that's my first question. And the second question is related to the lysine variant. So whether you did an experiment with the treating with CPB with and without. So what was the observation?
To answer your first question, sir, we have kept the same gradient program for all the 4 monoclonal antibodies. While using the pH gradient elution mode, we generally required a different gradient program. But in this particular study, with the same gradient program, salt gradient, I mean to say, we could able to achieve the good resolution amongst the different charge variants for all the 4 monoclonal antibodies. And second, to answer your second question, we have not studied the lysine truncation part. So only the without treating, I mean the samples were as such analyzed.
Okay, thanks, Ashutosh.
Thank you, sir.
So were the results, I'm Shafali here, were the results comparable to the results that you would get on by LBA? And were you able to do like all the experiments, or did you do only precision and accuracy? Were there also matrix interference or stability? Would it be feasible to do all the studies on this?
Ma'am, to be honest, we have not compared the result against the LBA. However, the results which were received at the customer end, those were comparable. Basically the pattern of the charge variants, but we have not compared our results with the LBA technique.
Great.
All right. So, if no further questions, then would like to thank you, Ashutosh. Thanks for your presentation.
And do you also do ADA on this? Anti-drug antibodies for these large molecules, for these MABs? Have you tried doing that?
No.
Great, thanks.
Thank you.
All right. So thank you, Ashutosh. Thanks a lot. And now let me invite our next poster presenter. So yes. So now we have poster number 6, Ritu Kamble, and she's going to present a poster on exploring the conformational changes and assessment of susceptible deamidation sites in stress-induced MAB. So over to you, Ritu. You can share your screen.
Yes, sir. Sir, is my screen visible?
Yes, and you have to do the slide share. I mean, yeah.
Yeah. So, good morning, everyone. My name is Ritu Kamble. I'm the student of MTech from Institute of Chemical Technology, working under the guidance of Dr. Ratnesh Jain and Dr. Prajakta Dandekar Jain. My title of the poster is Exploring the Conformational Changes and Assessment of Susceptible Deamidation Sites in the Stress-Induced Therapeutic Monoclonal Antibody. Let's get introduced to the topic. Protein-based pharmaceuticals are one of the fastest growing categories of therapeutics. However, they are prone to undergo degradation during various phases of manufacturing, such as production, purification, transportation, storage, or even formulation. This mainly happens due to stresses such as mechanical stress, pH, changes in pH, salinity, temperature, concentration, or chemical denaturation, or even misfolding of the protein. These stresses can induce modifications which may impact on safety and efficacy of the therapeutics. To control this problem, absolute understanding of the behavior of therapeutic proteins under different stress conditions is necessary. Thus, it is necessary to characterize them with suitable analytical techniques which help in analysis of changes in molecular weight, conformation, size, shape, and the state of the protein. This is the overall workflow of my work. My work aims to understand the physicochemical behavior of therapeutic protein trastuzumab biosimilar under stress condition as forced deamidation. So firstly, we titrated the protein solution with tris base till pH 9.2, then incubated it for 7 days at 37 degrees Celsius for the deamidation reaction. Then the deamidated protein solution was analyzed firstly by using LCMS for the identification of modified sites, then for stability testing with nano DSC. Then it was analyzed for presence of size variance by using AUC and particle size analysis by using DLS. Moving on to results. First, LCMS. We have identified the amino acid site-specific modifications of deamidation by using peptide mapping approach. And the sites we got are at N55, 84, 289, 318, 364, 387, and at Q82 in heavy chain and in light chain at N30 with respective relative abundance shown in the bracket. These are some of the method specifications used. N30 and N318 are most susceptible deamidation sites that we got. These are some of the graphs from LCMS. This is a overlay of extracted ion chromatogram of peptide maps of trastuzumab. Then you can see the chromatograms. In the chromatograms, that the modified peptide elutes later than that of the unmodified peptide, and in the MS1 spectra of the same, it has 0.329 Dalton mass addition in the modified peptide, and in the same peptide at +2 charge state there is addition of mass 0.49 Dalton at Y13 ion. So like this, we have confirmed the deamidation sites obtained here. Then next, we have performed nano DSC. So this graph shows the comparison of melting temperatures of the control and deamidated map. The melting temperatures for the both of the melting temperatures have been increased in the deamidated map, showing the presence of high molecular weights or aggregates, which generally take higher amount of heat to melt. These are some of the method specifications. Moving into dynamic light scattering. This is the graph of comparison of particle sizes of control and deamidated map. As we can clearly see here, the particle size of the deamidated map has increased, showing the probability of presence of high molecular weights. And due to the highly basic pH of the sample, there is negative value of zeta potential is also observed by DLS, indicating there is increased instability in the map solution. Then next is analytical ultracentrifuge. These are the CS distribution graphs of the control map map and a deamidated map. And the results obtained from the same are in the control map, there is in the deamidated map, there is presence of impurities such as aggregates at 0.039% and fragments at 6.8%. And we can conclude here that the map purity has been affected due to the formation of such aggregates and fragments or impurities. And in overall conclusion, the quality of the drug product decreases due to deamidation, leading to formation of impurities, which can further alter the activity of the drug. This provides the evidence of the need to establish a retest period for the active substance or revise the shelf life and recommended storage conditions for the finished product as per the guidelines. Then the future work can focus on understanding the cellular mechanisms driving these changes for calculating its effects on the efficacy and activity of the drug. These are the references. Acknowledgement. My sincere thanks to Ms. Dipti Bhandarkar from Shimadzu for helping me with LCMS analysis. I would express my deep gratitude to my supervisors for their valuable support and guidance. And I'm thankful to Amita Puranik for helping me with studies, and entire nanomedicine research group and my University Institute of Chemical Technology. Thank you.
Thank you, Ritu. If you can stop sharing now so we can highlight your visuals. Thank you. Over to the judges. Any questions for her?
Yeah, Ritu,
Go ahead, Ravi.
yeah, Ritu, a good presentation. So 2 questions. One, why you are calling this as a deamidation? Because when you subject it to a stress condition like heat, there is multiple possibility of formation of many post-translation modifications. So my first question is why you are specifically saying this as a deamidation? That is my first question. And the second question is why you haven't tried other modification, the stress conditions, other stress condition, as you rightly mentioned in the presentation, why only focusing on the temperature? You should have tried other conditions, and why you haven't measured other post-translation modification kind of demethylation, acetylation, and other aspects. Yeah, sorry, I have asked 2 to 3 questions. Yeah, please.
So firstly, deamidation is a very important post-translational modification. It has role in regulating heterogeneity and stability of the map. Then secondly, deamidation, I'm calling it deamidation because I've used specific protocol for it to keep it at high temperature and at high pH. The supposed modification is deamidation. Yes, we have also observed other modifications during LCMS analysis, but our main focus was deamidation in this case. And
other stress conditions?
Yeah, other stress conditions. So I said that deamidation, we are focusing on deamidation for this particular study. So
okay, the question is why you are focusing on the deamidation? Like I meant to say, because PTM per se should have all the modification. You need to check with any maps when you look for a biosimilarity perspective and a characterization perspective. Why you are specifically focusing on a deamidation?
Sir, as I said earlier, deamidation helps in, it's a mainly focuses it on regulating heterogeneity and stability of the map, and the asparagine and glutamine molecules mainly get affected in deamidation, which help in stability of the structural stability of the map, as well as some amount of functional stability. So we focused on that.
Thanks, Ritu. Good work. Keep it up.
Thank you.
Ritu, there is one question. This is Amarnath.
Yes.
I just wanted to know, have you worked your own on the DLS system, Gitasizer?
Yes, sir.
Okay, so
I'm
what are
taken
the
help for
common
LCMS.
okay, so what are the common challenges when you are developing a method for a particle size analysis on a Gitasizer?
Uh,
Did you come across with any kind of challenges when you are measuring the particle size?
yes, sir. As you can see, as you have, I mean, in the graphs that the particle size of the control map was slightly lower, that is 5 d nanometer, and in the literature, it's mainly 10 to 11 nanometers. So that is the thing I observed during during DLS analysis. But in method development as such, there was no any problem. So,
Okay, no problem.
yeah.
Thank you. I don't have any more questions.
Thank you.
Yeah, Deepak, over to you. Think. Deepak, you are in mute, I hope. We are not.
Yeah. Are you able to hear me now? Yes.
Yes.
So we have our now next poster presenter, poster number 7. The presenting author is Marianne Saldana. She's going to talk about extracellular metabolite profiling for LCMS for bioprocess optimization. So over to you, Marianne. You should be able to share your screen.
Yes, sir. Good morning. I will share my screen.
Yes, please. Yeah.
Yeah, is my slide visible?
Yes, very much. So please proceed.
Okay. Good evening. Good morning, everyone. I'm Marianne Saldana from the Institute of Chemical Technology in Mumbai, and I will be presenting my a part of my PhD work, which is on extracellular metabolite profiling by LCMS for bioprocess optimization. So, as an introduction, I would like to just give a brief about the production of monoclonal antibodies in CHO cell culture. People might know that it is a bit of a tricky process, and because there is a very large amount of nutrients in culture which keep on fluctuating in concentration, and this level of concentration of nutrients in culture is very important for cell growth and consequent map production. So it is important to monitor the levels of compounds during the culture period, and this can be an indication of the health of the culture. One such technique is mass spectrometry, which overcomes the limitation of detection in as compared to many other methods, and is also unparalleled in accuracy and specificity. So in this presentation, I will be showing the use of an LCMS method for extracellular extracellular metabolite profiling in day-wise cell culture samples. So which can be used to monitor the trends of nutrients present in the culture, and this such a method can be effectively used to monitor and improve bioprocess conditions and even modulate some CQAs. So the overall schematic of the study undertaken herein is where I've used two cultures, which one is in batch mode, two in batch mode and two in fed batch mode, with varying concentrations of glutamine and with varying concentrations of glucose along with a commercial feed. So from these cultures, I have studied the cell growth, the map production, and as well as the extracellular metabolite profile, which is basically spent media analysis, which is commonly known as. So from this nutrient pool in the culture, we have detected the whole range of amino acids, vitamins, organic acids, and including TCA intermediates, membrane precursors, a few of them, some nucleic acid derivatives, and some waste byproducts. This is a brief of the extracellular metabolite profiling method by LCMS. The method was performed on an LCMS 86T triple quadruple mass spectrometer by Shimadzu, coupled to an Nexera N-series UHPLC also by Shimadzu. The column was a reverse phase column. The details of the column are proprietary with Shimadzu, so I couldn't reveal it here. Mobile phase used was, since it was a reverse phase method, we used the regular water and ACN with 0.1% TFA. The method package used was called cell culture profiling method package, which is developed and owned by Shimadzu, which involves, in short, a flow rate of 0.35 ml per minute with a runtime of 17 minutes. In the 17 minutes, all the components, which is approximately 85 plus components which is present in the cell culture media, are analyzed. And in terms of sample preparation, proteins were first precipitated because these cell culture samples contain antibodies which are produced by CHO cells. So these antibodies and any other proteins have to first be precipitated out using ACN, and they were then diluted 10, 100, and 500 fold with water. Multiple reaction monitoring or MRN transitions were scheduled around the expected times in both positive and negative ionization mode with the help of standards. And the same standards were also, standards for each of the compounds were also used for preparing calibration curves, which were used for estimating the concentration of each compound. So this is a brief of the, a summary of the cell growth and antibody production results. So as you can see, you might know the difference between a batch and a fed batch, where extra nutrients are added in a fed batch culture. So those two cultures lasted for longer periods. And when glucose was additionally added along with the feed, it lasted for 2 more days, up to 11 days. And in terms of antibody production as well, the fed batch cultures lasted for longer. And when glucose was additionally added, even more antibody was produced as compared to the control fed batch culture. This is a summary of the extracellular metabolite analysis results obtained from the LCMS. This is only a representative of a few compounds because we have detected and quantified approximately 85 compounds. So for the purpose of just representation, I've picked a few from each of the prime categories. And just to show the various kinds of trends obtained over the culture period among the 4 different conditions which were studied. So we have some of the amino acids can give us very important information like ornithine, tyrosine, lysine, which were very rapidly depleted during the culture period. And this trend was almost similar in all the 4 batches. We have some amino acids like alanine and glycine, which had mixed profiles of first secretion and then consumption. And some of these trends varied between batches as well. And we also detected a whole range of vitamins, some of them including pyridoxine, which was completely depleted by halfway through the culture, implying that it's very critical for cell growth and probably needs more supplementation. Some of the other vitamins like niacinamide and nicotinic acid and folic acid were somewhat on the increasing side or fairly stable, and indicating that they were supplied in sufficient amounts and don't need further supplementation. Organic acids is a very critical and interesting category of compounds in this study. They give us an indication of the metabolic state of the cell. We can see from some compounds like citric acid and succinic acid, where we see a shift from secretion and then followed by consumption. This is basically due to a shift from the exponential phase of cell growth to stationary phase of cell growth, where the
Marian, sorry to interrupt. You have 30 seconds. You can conclude.
Okay,
Your time is
sure.
up. Yeah, thank you.
Yeah, so the metabolism shifts from glycolysis to oxidative phosphorylation. So such analysis can give a very a brief and interesting indication of cell growth and metabolism. This is just a summary of all the amino acids which were quantified in different cultures. And we can see that 4 amino acids, glutamine, asparagine, serine, and lysine, were consumed to more than 90%, indicating that they need to be supplemented in feed. And in conclusion, from these results, we can determine which are the best candidates for supplementation in feed. In this case, it was glutamine, lysine, serine, asparagine, ornithine, etc. We found a clear shift from glycolytic metabolism to oxidative metabolism in stationary phase. And this is a good indicator for studying metabolism. Finally, I would like to say that such a study on LCMS can effectively to design superior feeding strategies, which can help in enhancing cell growth and MAP production. Thank you.
Thank you, Marian. Thank you for your presentation. Over to the judges. I think there's already one question for you in the chat box. So if any of the judges would like to take up that.
Yeah, sorry, am I audible?
Yes, very much. Please proceed.
Yeah, so it's really a great work which would serve for initial evaluation of growth and protein concentration purpose. So my one question is whether how many methods you have used to analyze this so many components? Is it 3 to 4 methods, because each analyte would need a different chromatographic condition as well as a different set of columns. So to analyze all the components, how many methods you have used?
So this is one single method. It was developed and owned by Shimadzu. It's called a cell culture profiling package, which they have developed. So it's just one single method. In a 17-minute run, we can detect all amino acids, a range of vitamins, organic acids. So basically, you just need a standard, and you'll need to set your MRM transitions beforehand, and set up a calibration curve, and you can detect it. It's just one single method. There are no multiple methods used here.
Okay, that's great. Then my second question is, all the analyte, the required limit of quantification has been met, because certain additional component what we generally add, the concentration would be very, very minimal during the growth cycle. So whether the required limit of quantitation has met.
Yes, so that is one limitation. Some components, we set up an LOQ based on the calibration curve, based on an R square that we get. So any concentration in that compound which does not fit in the calibration curve is not used. So that compound will not be analyzed or quantified. So we have carefully selected only compounds that can be reliably detected and quantified using the calibration curve.
Okay, I think a great presentation, and this would certainly help for any biologic manufacturer. So my only recommendation is please, improve this method to the lower limit of quantitation using high resolution mass spec and other part, and this would certainly help. A good presentation.
Okay. Thank you, sir.
So I think one question is there in the chat box from Soumitra Gajjar. I know the answer, but why don't you take up this? Can amino acid analysis be done by HPLC-based method using FLR detector, please?
Yes, it can be done. There are a lot of commercial kits available to be done, which facilitate its detection by HPLC, which is fluorescence-based. In this case, we didn't need any fluorescence tagging because the detector used here is mass spectrometry. So, but you can do just amino acid analysis by HPLC. However, here we have used a combined method which was developed by Shimadzu in a mass spec based method. So we didn't need a fluorescence detector here.
All right, so if no further question for Marianne, then thank you, Marianne. And let's proceed further with the next presentation. So now we are on poster number 8. Navya Ajit Kumar Bhaskaran is the presenting author, and the poster title is the reverse phase HPLC method development and validation for rapid and simultaneous estimation of hydrophilic and lipophilic drug encapsulated in a nanoformulation. So over to you, Navya. Feel free to share your screen.
Yes. So is it visible? I would just like to know that.
Yes, it is visible. It is visible. You're good to go, please.
Okay, thanks. Good morning to one and all. I'm Navya. I'm from the Department of Pharmaceutics, Manipal College of Pharmaceutical Sciences, MAHE. I'm here to present my work entitled A Reverse Phase HPLC Method Development and Validation for the Rapid and Simultaneous Estimation of a Hydrophilic as well as a Lipophilic drug, which is encapsulated in a nanoformulation. I would like to give a brief introduction to the work that has been conducted. Irinotecan is a topoisomerase inhibitor which is used in the treatment of cancer, primarily colon cancer as well as rectal cancer. One of the major side effects seen with the current marketed preparation is the myelosuppression, diarrhea, neutropenia, and anemia, which in certain cases have led to the patient from withdrawing therapy as well as death in certain cases where it is really severe. The problem which we identified here was irinotecan undergoes P-glycoprotein efflux in the intestine as well as colon, and it is the hydrophilic drug which has been mentioned in the title of the work. And since it undergoes the P-glycoprotein efflux, there is a low bioavailability of the drug at the site of action, which is around 9%. Quercetin is a hydrophobic drug, and it is a well-known flavonoid which exhibits P-glycoprotein inhibitory action. So we observed a possible solution to the problem which is seen with irinotecan, which is formulating both the drugs into a nano formulation, which would help in overcoming the problem of P-glycoprotein efflux. So there was an analytical problem in this, that there is no specific analytical method for the simultaneous estimation of both the compounds in nano formulations, which is why we have developed this method. So the method conditions are as follows. It was an isocratic method. We used the Luna C18 column. The mobile phase was a 25 millimolar ammonium acetate buffer, which was adjusted to a pH of 2.5 with trichloroacetic acid along with methanol. The detection wavelength was a UV detector at 254 nanometers. And we observed good separation with a retention time of 9.5 minutes for irinotecan and quercetin at almost 13 minutes. So from the chromatogram, we observed that there is no much interference from the mobile phase, which shows that it is specific for both the drugs. The system suitability parameter showed that there was not much variability in the percentage CV for both the drugs for the responses of retention time and peak areas. The linearities were also showing a good R-square value for the individual drugs of 0.999 and 0.9998. The limit of detection and limit of quantification for irinotecan was found to be 63 nanograms and 306 nanograms for quercetin, while the LOQ values for irinotecan was 212 and 1020 for quercetin. Moving on to the precision and accuracy studies. The precision studies were done as an intraday and intraday precision study. The intraday precision studies showed percentage series values within 1% for both the drugs, and the intraday precision studies showed the percentage series within 2%. The accuracies also for irinotecan, we saw the percentage accuracies were all around 99 and 100%. While for the this was a standard addition method which was followed. The accuracy values for quercetin showed a head-to-head comparison when it was done for the concentrations for the pre-mix as well as the post-mix samples. It showed a good correlation. The robustness studies were done wherein parameters such as the injection volume, the detector wavelength, the column oven temperature, and the flow rate were carried out at different values. And we observed that the percentage CV values for the retention time and the peak area for both the drugs were well within the limits. The application of this study was mainly done to calculate the drug content of irinotecan as well as quercetin in the polymeric nanoparticles which were formed. So we prepared these polymeric nanoparticles using PLGA and bioprobe sonication method. We did the characterizations using particle size, zeta potential, and polyviscosity index using the Malvern zeta sizer. And we have used this method for the estimation of the total drug content as well as the encapsulation efficiency of both the drugs within these nanoparticles. So we obtained the results of 86% of total drug content. There were some drug losses for irinotecan, and quercetin was around 98%. And the entrapment efficiency was 41% for irinotecan and quercetin was 55%. So I would like to conclude saying that we develop a sensitive, precise, and accurate method for the simultaneous estimation of irinotecan and quercetin in the nano formulations. And this method would be helpful for researchers for their quantitative analysis of both the drugs in nano formulations. I would like to thank ICMR for providing financial assistance and Laura's Labs for providing a gift sample of the drug, as well as my college and my guide for permitting me to work on this project. These are my references. I thank you for your attention.
Thank you, Navya. You can now stop sharing the screen so we can display your. Yeah, so over to the judges for any questions.
So Navya, I have a quick. Yeah, go ahead, Amarnath.
Okay. So Navya, this is Amarnath. I have just one question. Irinotecan, it's a liposomal product. Have you also looked at the leakage of the encapsulated product?
Sir, leakage as in I didn't understand.
So liposomal leakage, I mean, there is a tendency that liposomal get breaks during, sample prep or, the solution preparation. And there is a possibility that the free component can also be, the leaked from the liposomal drug product. I mean, this is the typical tendency of any kind of the liposomal products.
Yes, sir. Actually, that's why I think the entrapment efficiencies that has given, we have used an indirect approach for calculating the entrapment efficiency. So we calculated the free drug and we know the total drug content. So we have subtracted the two and then we have calculated the entrapment efficiency. So there has been either leakage or some drug losses during the procedure.
So how would you ensure that there is no further leakage during your entire sample prep?
I am not sure of that.
Okay,
sir.
no problem. That's fine. Go ahead, Jignesh.
Yeah. So Navya, good work and a novel approach to improve the bioavailability of poorly absorbed compound. So I have two questions. Considering the hydrophilic and lipophilic compounds, how did you arrive to this chromatography, mainly the column selection? Because you achieved a reasonably good retention for both the compound.
Yes, sir. Actually, it took me quite some time. We had to screen many columns, and I did a literature search, and in that I happened to find a method where both the drugs were separated using the mobile phase, but there was a problem of the stationary phase. However, I tried many methods, and when we were using ACN as the organic solvent, this was not, we were not getting a good separation Mostly the drugs would adhere almost close to each other, but using methanol had a great impact on the separation. I did not look into why that has happened, but this was what we observed.
Okay, great. And the other question is that since the application would be for formulation part,
yes,
and you applied extraction method using DMSO,
yes.
So have you checked any interference from the placebo?
Sir, there was no interference. We had given a blank formulation also in the similar pattern, and there was no interference. The mobile phase as such was not having any interference at the retention time of the drugs. So I don't think there should be a problem.
Okay, thank you.
All right, so if no further questions, then would like to thank you. And let's move on to the next presentation. Thank you, Navya. Let me quickly display. Yes. So now our next presentation is poster number 9. The presenting author is Shikha Patel, and she's going to talk about force degradation. Okay, one second. Yes, force degradation study of peptides. So over to you, Preeti. And Preeti, if you have switched on your video. Yes.
Yeah. Am I audible, sir?
Yes, you are very much audible, and
my screen is also visible, right?
Yes, please proceed.
Okay, thank you. I'm Ms. Shikha over here from the Department of Pharmaceutical Analysis, Institute of Pharmacy, Nirma University, and I'm going to talk about force degradation study of peptides. So why I have selected to work over the peptides? The peptides are actually defined as an alpha amino acid polymers with a specific defined sequence having 40, I'm so sorry, something has happened. Yeah. 40 amino acids or fewer in size as per the regulatory agencies. These have a diverse chemical modifications, advancement in the drug delivery system, and have a very good bioavailability and very good clinical significance in the acute and chronic metabolic disorders. So I have chosen to work over the peptides. Basically, the peptides are of 4 different types depending upon the differentiation from the endogenous peptide from where they are belonging. That can be native, analog, heterologous, or peptide conjugates. If we look towards the wide variety of the formulations available in the market, so they are available in the form of starting from the injectable to that of the oral, as the stability of the peptides are very promising. So I have decided to work over the force degradation study. Force degradation study is nothing but the intentional degradation of the active ingredient as well as the drug product under the various stress conditions of pH, temperature, light, and oxidizing agents. They are very promising and predominant in the determining the intrinsic stability of the drug substance when designing and optimizing their formulations, developing the specific, robust, and sensitive analytical methods, and it very much helpful in determining the specifications of impurities based on their toxicity characterizations. So moreover to that, force degradation studies can also be helped to understand the critical quality attributes of a molecule by highlighting the degradation routes. Am I audible, sir?
Yes, very much. Please continue.
Okay. What we need to look towards when we are working with the force degradation studies of the peptides. Peptides are regulated as a small molecules by the regulatory agencies. So it's very simple. If we know about the structure of and the amino acid sequence of the peptide, then we can pretty much determine about the type and concentration of the stressor that we can use. These are the commonly stress conditions employed for the force degradation studies of the peptides. They are evaluated as per the strategy of ICH guideline Q1A, Q1B, and Q2B, similar to that of the small molecules only. Commonly known degradation pathways for the peptides, they are different than that of the larger molecules as well as the small conventional molecules. Majorly the deamidation of the aspartate and aspargine and aspartic acid is there, along with the glutamic acid and glutamate. Isomerization of aspartic acid is very much common. Beta elimination of the cysteine, serine, and threonine. Oxidation of the methionine, histidine, tryptophan, tyrosine, phenylalanine, and cysteine. These are the most promising amino acids prone to the oxidation. And there is also polymerization at these sites. These are the commonly known amino acids which are prone towards the different degradation pathways. So moving towards the work of mine, that is, I am working over the post-degradation study of cetrorelix. Cetrorelix is a decapeptide with amino acid sequence elaborated over here. If we look towards the sites which are promising toward the degradation, then the serine, tyrosine, these are the amino acids which can undergo the degradation extensively. So this is the chromatographic conditions which I have employed for the post-degradation study. I have worked over the UPLC with a PDA detector. It is made up of waters with Empower software. The mobile phase A and B I have chosen in such a way I want to make the method compatible with the LCMS part, as I have to do the mass analysis also. So this is the mobile phase. Diluent I have used methanol is to water. Common column I have used, that is C18 with 254.63 micron. It is an isocratic mode with 63:37% ratio. Flow I have kept as 0.8, wavelength is 226. The total runtime, that is 30 minutes, and retention time of the cetrorelix is about 14 minutes. Optimized stress conditions, that is, it is too much, as the cetrorelix is containing serine, it is too much degraded under the alkaline conditions. No amino acids are that much of prone towards the oxidation, so no degradation has been observed in the case of oxidation. Definitely there is a tyrosine which can be prone the molecule towards the light degradation. So these are the optimal conditions with the percentage degradation observed. If we look towards the chromatograms, this is for the cetrorelix. This is for the thermal degradation. There is no degradation observed over here. In the case of acidic pH, yes, we are getting 3 degradant products. In the case of base, it is extensively degraded with different degradation products. In case of also peroxide, there is oxidation. There is no oxidation happening over here, so there is no degradation observed. But in the case of UV, yes, we are getting some sort of degradation. So this is the common degradation products which are summarized over here, along with the stress conditions and relative retention time. The possible degradation can be happen, that is the, as we are looking towards the this two degradation products which are very closer to the principal peak, so there is a possible of generation of the enantiomers. Serine is there, which is more prone towards the degradation, can be hydrolysis or can be beta eliminated.
Shikha, sorry to interrupt, but time's up. So in 30 seconds, you can conclude. Thank you.
Yes, yeah. So as there is a terminal amide group that can be converted into the carboxylic acid under the hydrolytic conditions, tyrosine can also undergo the oxidation and then can be possible of peptide bond cleavage. Future plan, that is as the drug is degraded too much under the alkaline condition, I need to confirm the degradation products by the LCMS characterization so that I can portray the degradation pathway. Along with that, the method is also need to be get validated for the employment of the same for the formulation products. Thank you so much.
Thank you, Shikha. Over to the judges. Any questions? So one question is, Shikha, how did you actually zero down on the mobile phase and the stationary phase for this? I see you use a TFA, but did you try using other mobile phases also?
Actually, my primary aim is to develop an analytical method which is compatible with the LCMS, so I have started with the TFA itself. Yes, I have used 0.1% TFA in acetonitrile as the mobile phase B also, but there the peaks are not retaining too much, they are eluting faster. So as you have seen in the acidic chromatogram also, the peak is eluting at around 4.5. So in case of 0.1% TFA in ACL, the peak is almost eluting near the void volume. So I have skipped that part and I have modified my mobile phase B towards the acetonitrile is to water.
Okay. Any other question from any of the judges?
Yeah, one question from my side, Ravi. So, so you have identified in the Cetrorelix case couple of impurities,
Yes.
but it has not been identified. I mean to say, it's like
Yes, yeah. Just I have separated them.
Yeah, correlation with the proposal. So your theory is perfectly all right. What are the vulnerable amino acid that would go for a modification kind of a thing. However, to prove this, that's really complex and challenging. So is it your next stage, or it's a part of your work?
Yes, I'm planning to analyze my stress sample by the ion trap analyzer or by the QTOF for the determination of the molecular mass of the degradation products, and confirm the same by the tandem mass so that I can come with the conclusion.
Thank you, Shikha.
Okay, sir.
All right. Thank you, Shikha. Thanks a lot. Let's move on to the next one. Okay. So, all right. So maybe some background was coming from Shikha once you completed your presentation. Yeah, but now you're fine. Okay, great. So let me just go back to the schedule. So now we are at poster number 10, and the presenting author is Arpit Bana. He's going to talk about charge heterogeneity analysis of selected biosimilar formulations using imaged capillary isoelectric focusing technique. So let me invite Arpit.
Hello. Hello.
You are audible.
Okay, sir.
Yes, you are audible. And, yeah, your screen is visible. You have to make a slide share mode. And if you can, yeah, your video is on already. Great. So yes, you are pretty much visible also.
Okay. So is it visible in slide mode?
No, you have to do it in a slide mode now, I guess.
Okay, so it is not visible. So now is it visible?
For some reason it is not, but in interest of time, let's continue. We are able to see your, just once you can click this slide share mode. One more time, the slideshow mode, if it is happening.
Yes, sir. Yes, sir. Let me try. Now is it visible, sir? In slideshow.
Perfectly okay.
Okay, sir.
Yeah, you can go back to the first slide and you can then start.
Yes, sir. Okay. Good morning to all the judges and panelists. Myself, Arpit Bana. I'm a PhD scholar at Department of Pharmaceutical Analysis, Institute of Pharmacy, Nirma University. I'm doing a PhD under Dr. Preeti J. Mehta, who's a professor and head at Department of Pharmaceutical Analysis, Institute of Pharmacy, Nirma University. So today I'm presenting a part of my PhD work. So title of today's presentation is Charge Heterogeneity Analysis of Selected Biosimilar Formulations using the Imaged Capillary Isoelectric Focusing Technique. So the flow of my presentation, it will include the aim and objectives, charge variant analysis and ICISF technique, experimental studies, charge variant and isoelectric point comparison, graphical representation of the data obtained, statistical analysis of the data obtained, study summary, acknowledgement, and references. So aim and objective. So my aim was to compare the charge variant profile of selected biosimilar formulation manufactured and marketed in India with their innovator. So the objective is to perform the comparative charge variant analysis of biosimilar formulation with the innovator product using the image capillary isoelectric focusing technique. So to find out the percentage similarity in the acidic, neutral, and basic variants of innovator and biosimilar formulations, and to compare the isoelectric point profile of biosimilar formulation with the innovator. So some definition, the innovator product is the biological product first manufactured by an innovator and later approved by the regulatory body is called an originator product or the innovator product. The biosimilar formulations are the biological product highly similar but not identical to the originator product approved by the regulatory body is called a biosimilar product. So what is the charge variant analysis and ICSF technique? So therapeutic proteins are complex molecules produced by living cells that treat a range of diseases. Many variables and cell culture conditions such as temperature, pH can lead to protein modifications such as oxidations, deamidation, lysine truncation. These modifications can make the protein more basic or acidic in nature, thus results in a protein population that is heterogeneously charged. Small variations in the charge of the protein can reduce drug efficacy or cause an immune response in the patients. Therefore, controlling the charge variant is critically important for product quality and safety. So now we are moving to the experimental study. So innovator and biosimilar formulation charge heterogeneity analysis were performed using the ICSF technique on Morris instrument by Protein Simple. So therapeutic map having different formulations were evaluated. Total 3 samples were evaluated. So there is the innovator product and there is biosimilar 1 and biosimilar 2. All 3 of them as having the concentration of 25 mg per ml. Now moving to the sample preparation. So first of all, for whenever we are performing an capillary restrictive focusing technique, preparation of the master mix solution is required. Along with that, the sample preparation is required. So for the preparation of the master mix solution, the methyl cellulose, urea, immunodiacetic acid, arginines, and pharmalites, as well as the PI markers were optimized. So the PI marker of 6.14 and 9.5 was selected as according to the isoelectric point of our selected in a therapeutic monoclonal antibody. Methyl cellulose was optimized for the viscosity as well as for the improve the separation. Urea is further optimized for the unfolding of the protein, as well as the immunodiacetic acid and arginine are further optimized as the cathodic and anodic stabilizers, and pharmalites, near range pharmalites are used for the separation. Okay,
Arpit, are you there? Arpit,
160 microliter on the master mix solution.
Okay, so we
am I not audible?
Just for I think 15 seconds we lost you. So I would request you can switch off your video, no worries. So maybe the bandwidth issue can be, they could be on your side. I'm seeing it's a less bandwidth. So you can switch off your video and you can continue. Yeah, please.
Okay, sir.
We can hear you now, though. You can continue, please.
Okay, sir. Okay. So again, the initial concentration was 25 mg per ml, which is diluted to 2 mg per ml. And then from that 40 microliter of the solution was mixed with 160 microliter of the master mix solution. And thus the final concentration of the sample was 0.4 mg per ml. Now, these are the electrophysiograms of the innovator biosimilars that are obtained. So as you can see here, we can see the standard markers of 6.14 and 9.5. And then we obtained separations of acidic variants as well as the basic variants in all the formulations. So 2 acidic variants as well as the 2 basic variants were separated along with the monomer peak. So the PI range is from 7.3 to 8.268. Now this is the overlay of the innovator and the biosimilar electropherograms. So as you can see, the acidic variants are separated along with the basic variants. Now charge variant and PI values comparison. So this is the table showing the comparison of the area percent of the charge variants. So there is we have divided the charge variants into the acidic variants, main peak, as well as the basic variants. So as you can see from here, we can see that the highest percentage of the acidic variants is for the innovator, and the lowest is for the biosimilar one. Similar, and for the main peak, the highest is for the innovator, and the lowest is for the biosimilar one. And for the basic variant, the highest basic variant was observed in biosimilar one, while the lowest basic variant percentage was observed for the innovator. Similarly, for the PI value comparison, so PI value comparison was done for the according to the peaks: acidic peak 1, acidic peak 2, main peak, basic peak 1, and basic peak 2. So from that, we can observe that when we are comparing both the biosimilars with the innovator, there is a slight variation, but it is within the limit. Now, this is the graphical representation of the data that is obtained. So as you can see here, with the control of our biosimilars, that some deviations are there, but they are within the limit for the PI also, as well as for the main peak, SAD peak, and basic variants also. Now, the statistical analysis of the chart variants was done by for using the GraphPad Prism software, and one-way ANOVA with the post hoc Dunnick test was applied, and the p-value was considered as a signifying parameter, where p-value less than 0.05 representing significant difference. So this is the one-way ANOVA table data for the acidic variants, main peak and basic variant, showing that the biosimilar 2 is showing less significant variation with the innovator, while the biosimilar 1 is showing more deviation with the innovator. Now, similarly, the statistical analysis of the PI values is also there, where we can see that there is no significant deviation.
Yeah, sorry to interrupt, but time is up. So you have 30 seconds, so you can conclude. Thank
Yes,
you.
sir. Yes, sir. Yes, sir. Similarly, for the PI values, there is no significant deviation was observed between the biosimilar 2 as well as the innovator product. So the studies that ICF profile of therapeutic map biosimilar market formulation was developed and was compared to the innovator. And the ICF profile biosimilar formulation with the innovator, it is evident that basic variants are high in biosimilar formulation compared to the innovator product. And similarly, the concentration of acidic species is high in the innovator and biosimilar 2 formulation compared to the biosimilar 1 formulation. Finally, after the complete ICF study, we found out that the innovator and biosimilar 2 formulations are more similar to each other compared to the biosimilar 1 formulation, and having high levels of deviation with respect to acidic, main, and basic areas and PI value. Difference in the peak height of the innovator and biosimilar formulation required further clarification data to support this deviation in the peak height. So for the identification and characterization of this modification is required to be carried out using the LCM cutoff. So acknowledgement, I would like to acknowledge Biotechno India for providing the facilities and resources for carrying out the project and the R&D center in Pune, as well as I also like to acknowledge Mr. Manish Singh and Hitesh Sharma for their help and support, as well as Mr. Rakesh Panchal. So these are my references. Thank you.
Thank you. Over to judges. Any questions for Arpit?
Yeah, Arpit, Ravi here. So
yes, sir.
One important question. So
yeah, Arpit, now you can switch on your video so we can display your visual. Yes.
Yes, I have switched on my video.
So one important questions when you are representing the basic variant, be careful on that, because many a times lysine variant, if you mishandle the sample, you will get a different lysine variants, which may kind of a pseudo variants. Okay, so my question was whether have you studied CPB treated and without treated for charge variants? So
yes, sir. We have treated it, and we have seen that for the biosimilar formulations that are manufactured in India, after CPB treatment, the basic peaks are getting reduced. So it means that the C-terminal lysine variant is getting removed.
Okay. So you are claiming that whatever the basic charge variant is a true charge variant. That's your claim, right?
Yes, sir.
Okay. So have you done the comparative evaluation of your CIEF method with CEX method? Because that's the general way. Yeah.
Yes, sir. So it is in progress, sir. Right now, it is in progress.
Okay. Good. Okay. Good work.
Thank you, sir.
All right. So if no further question for Arpit, then I would like to thank you, Arpit, for your presentation. Let's move on, and we will go to our next presentation.
Thank you, sir.
So now we are on poster number 11, which is from Aditya Narvekar, and he's going to talk about pH-dependent aggregation and conformational changes of rituximab using success and its comparison with the standard regulatory approach of biophysical characterization. So Aditya, over to you. You should be able to share your screen. Yes,
Hello.
yes, Aditya, we can hear you, and you can do the slide share mode if you
Yes.
can do that. Yeah.
Is
Perfect.
it visible?
Yes, yes, you're audible, visible, and your slides are up in there. So ready, you can.
Okay. Good morning, everyone. I'm Aditya Narvekar, PhD student from the Institute of Chemical Technology, Mumbai. I'll be presenting our work on pH-dependent aggregation and conformation changes of rituximab using SAXS and its comparison with the standard regulatory approach of biophysical characterization. Understanding the rationale behind the work, biosimilars, as you all know, are anticipated as an affordable class of drugs, but there is a sizable section of patients in developing economies who cannot afford them. Therefore, we need to implement innovative strategies which would help to lower the development cost of biosimilars and make them affordable for all. Our work therefore focuses on one of the important steps in the development of biosimilars, which is physicochemical characterization. Now, this step of physicochemical characterization involves the use of several techniques in order to establish orthogonality in their CQAs. However, the use of so many techniques adds to the overall cost of the product and therefore becomes unaffordable in developing economies, example, our own country. Hence, we suggest the use of X-ray, small angle X-ray scattering as one of the techniques for identification of their CQAs during especially the batch release of formulations. The methodology adopted in this work involves conventional biophysical characterization for aggregate and structural analysis using the conventional techniques listed on the screen, and aggregate and structural analysis using SAXS. Beginning with the results of SAXS, the scattering data obtained from SAXS provides us with the P of R distribution of the antibody sample, which is obtained by the indirect Fourier transformation of the scattering intensity. The peaks here indicate differing shapes in each of the pH conditions, indicating changes in the overall solution conformation of the protein. The two peaks which are present indicate a distal correlation between the two folded domains in the protein, which is a characteristic of a flexible solution conformation. When we do quantitative estimation of these two peaks, the ratio of the intensities of the two peaks indicate that there is slight change in the overall solution conformation in the acidic condition, while there is very negligible change in the basic condition. Another parameter which we obtained by the indirect Fourier transformation of the scattering intensity is the radius of gyration. The radius of gyration values, if overestimated, indicate the presence of higher molecular weight products or attractive protein-protein interactions. Here we observe that the RG values are very much closer to the control, indicating negligible presence of aggregates or no presence. Similarly, the RG values obtained from other conventional techniques like SVAUC and DLS also indicate the similar conclusion. The other parameter which we obtain from the SAXS data is the mass fractal dimension. Proteins are considered to be mass fractals with a value of 2.5, and the mass fractal values obtained from the several techniques range from 1.4 to 1.5, which indicate that there is a presence of loose irregular aggregate, which basically follow diffusion cluster aggregation model. Other parameters which we obtain from the SAXS data include the Kratky plot, which is used for the qualitative estimation of the protein fold in the various buffer conditions. In the pH 6.5 and 2.2, we see that there are two parabola-like curves which do not converge to the x-axis, indicating that there is flexible solution conformation of the protein at these pH conditions. Whereas in the case of pH 10, there is an elevation of the baseline towards a hyperbole-like curve, which is because of the presence of a certain population of the protein behaving as a random call. When we do further quantitative estimation using Kratky plot, we obtain the Q value, which is the Porod invariant value, which is basically the integral of the area of the Kratky plot. From these values, we also quantitatively confirm that the pH 6.5 and 2.2 do not show much change in the overall protein fold, but there is a change in the protein fold because of the presence of random coil at the basic condition. The other parameter which we obtain from the Kratky plot is the S by V ratio. The S by V ratio indicates slight increase in the value in the stress conditions in acidic and basic buffers, indicating changes in the protein conformation because of the pH stress. Further, moving on to the Guinier plot, which is obtained from the Guinier region of the SA-XS data, obtained at low or intermediate Q values. The absence of linearity of a Guinier plot indicates that there is the presence of aggregates. However, we see here that the plots are almost linear in all the pH condition, indicating again the absence of aggregates or negligible presence of aggregates. So in conclusion, when we compare the conventional biophysical characterization using several techniques and the corresponding data obtained from them correlates with the data which is obtained from the small angle X-ray scattering technique, which provides us data from a single experiment with respect to Guinier plot, radius of gyration, mass fractal dimension, P of R distribution, and the Kratky plot. And thus, this technique therefore demonstrated a comparison between the SA-XS versus the standard regulatory biophysical approaches. They were comparable with respect to the inferences drawn and also observed to fulfill the collateral investigation which is required in regulatory approval, and therefore holds a tremendous potential to transform the field of biosimilar characterization and contribute towards accessibility of such therapies across socio-economic strata of the society. Thank you very much.
Thank you, Aditya. You can stop sharing so that we can display your video. Yes, over to the judges. Any questions for Aditya?
Yeah, Aditya, it's really a great presentation, and one of the very good work for comparison of aggregates with multiple orthogonal technique. That's the need of the hour. So, and application of the SACs is a kind of a new addition. Till now, we were working on multiple other aspects. So, one of the question from my side is, how do you justify the variability of aggregates? Even though you have mentioned, couple of aspects, but I just wanted to go in depth. Like, how do you justify the variability with different techniques?
So basically, the variability with different techniques depends on the interactions of the solute with the analyte which is being detected. So in the case of SEC HPLC, we see that the analyte interacts directly with the column, where therefore the sensitivity is reduced because of the use of pre-filters in a column, and therefore the aggregates estimated are not the true representation of the sample. But where the stationary phase and the analyte interaction reduces, such as DLS and SVAUC, we see that it is a true representation of the aggregate quantitation of that sample, as compared to techniques which involves the use of stationary phases. So that is the basic limitation and leading to the varying percentage of aggregates which we obtain across various techniques.
Okay, one last question. Have you studied a reversible and irreversible aggregate in the AUC? Because by multiple permutation and combination, you can do such kind of experimentation.
We haven't done that in this study.
Okay, but yeah, but excellent presentation and a very good work. Yeah, thank you. Keep it up.
All right, so if no further question, then thank you, Aditya, and we will move on to the next presentation. So the next poster presenter is Ditiksha Desai. She's going to talk about cyclosporine delivery from graphene oxide laden contact lenses for management of dry eye syndrome, in vitro and in vivo studies. So over to you, Ditiksha. You can share your screen.
Yes, sir. Are you able to see the screen, sir?
Yeah, you have to just do the slide show so that, yeah, perfect. You are good to go.
Okay. So good morning, one and all. I would like to present my research topic on cyclosporine delivery from graphene oxide laden contact lenses for management of dry eye syndrome, its in vitro and in vivo studies. To begin with the introduction, most of the ocular dosage forms available are eye drops or ointments, and they are to be instilled very frequently. So the limitations are frequent instillation. They have nasolacrimal drainage, because of which there is drug wastage. There are side effects due to high dose. It gives less than 5% viability and short precorneal residence time. Now, in order to overcome the limitations of conventional dosage forms, ocular dosage forms, several novel drug delivery systems have been explored, like in situ forming gels, nanoparticles, contact lenses, intravitreal or subconjunctival injections. One of the drug carrier available are the derivatives of graphene. Now, it has several properties, but the most interesting properties for the ocular delivery are that it has EMI shielding property, it has high mechanical strength, electrical conductivity, has large surface area for drug loading, and it prevents the contact lenses from dehydration. So the aim of this research project was to develop cyclosporine graphene oxide laden contact lenses to achieve controlled ocular drug delivery without compromising the critical lens properties, that is its percentage swelling and transmittance properties. Now, cyclosporine is an immunomodulator which is used in dry eye syndrome, that is keratoconjunctivitis sicca, which is desiccation of cornea and destabilization of the tear film. It is the first line treatment for this dry eye syndrome. So I have chosen it for my research purpose. The brief objectives are to synthesize and characterize graphene oxide, fabricate and evaluate contact lenses. The contact lenses will be fabricated by cast molding method, photopolymerization, and to perform the animal studies. The graphene oxide was synthesized using modified Hermes method, wherein the size of the graphene oxide was found to be 638 nanometers and PDI 0.180. Now, graphene oxide, it is a two-dimensional array of graphite of carbon atoms which are intercalated with the oxygen functionalities, that is carboxyl, epoxy group, hydroxyl group. So that is the constitution of the graphene oxide. Now, in order to confirm this conversion of graphite to graphene oxide, several spectroscopic and microscopic techniques were performed. So the results are shown here. From the first graph is the XRD in figure number 7. So from the XRD, it is revealed that the graphite shows a sharp peak at 2 theta of 26, which was diminished or which shifted to 10 to theta. Also, the distance between the two layers of carbon in graphite, that is 5 angstrom, which 3 angstrom, which increased to 8 angstrom in graphene oxide. Now, this is because of the intercalation of the oxygen groups in the graphite layers. Another is the Raman spectroscopy, which has a characteristic G band, which is shown, and it is due to sp2 carbon atoms which are present in graphite, and it has a very small D band. Now, this G band, the intensity of G band decreased in graphene oxide, and the intensity of D band increased, which also says that the graphite has converted into graphene oxide. Another is the FTIR studies, which showed all the characteristic peak of oxygen containing groups like epoxy. There is a broad peak of hydroxyl group around 3500 centimeter inverse. This is an HR-TEM image which shows that graphite is a crystalline material while graphene oxide is an amorphous material, in same as that shown in the XRD. The EDS elemental analysis was performed, and it revealed that there is 12% by weight oxygen present in graphene oxide in contrast to less than 0.5% in graphite. So all these techniques confirm that graphite was converted into graphene oxide. Now, the contact lenses, the drug loading on contact lenses was performed by two methods, soaking method and direct loading method. Now, in soaking method, the blank contact lenses were fabricated by cast molding method, and they were dipped into the cyclosporine solution of 10 microgram per ml. And in the second method, direct loading, the drug itself was added into the monomer solution of the contact lenses, and then the lenses were fabricated. Okay. Now, the control contact lenses are those which doesn't have drug, and it also doesn't have the graphene oxide. So it shows the percentage swelling and transmittance property greater than 90%, which are within the ISO standards which are required. Now, as we load the drug into the contact lenses, the percentage swelling decreases. Here it is 73, and in direct loading it is 53. Now this is because the drug cyclosporine is hydrophobic in nature. When the lenses are dipped into the solution, into the packaging solution that is simulated tear fluid, the drug precipitates within the lens and the lens becomes hazy. Now such hazy lenses will interfere with the vision. Now then there are different batches wherein the graphene oxide was loaded in different amounts like 100, 400 and 800 nanogram. And we can see that there is an improvement in the swelling as well as transmittance properties. Now, the improvement in the swelling is because of the hydrogen bond formation between the oxygen groups of graphene oxide and the water into which the lenses are dipped. And the improvement in the transmittance properties is because of the molecular dispersion of the drug onto the graphene oxide by pi conjugated spectra. So the similar results were obtained with the lenses of both the types, that is soaking and direct loading, wherein on increasing or on addition of graphene oxide, the swelling was improved as well as the transmittance was improved. If we talk about the assay, the drug uptake in case of soaking method is higher with the lens SMGO 400N, that is, it is containing 400 nanogram of graphene oxide as compared to without graphene oxide lenses. And in case
Ditiksha, your time is up. So in 30 seconds, you need to conclude. Thank you.
Okay. Even the contact angle, which should be less to improve, to give the improved wettability, on addition of graphene oxide, the angle decreased. This is an in vitro release from which we can say that as we increase the graphene oxide, the amount of drug release increases. Molecular docking studies were performed, which confirmed the interaction between the graphene and the cyclosporine, and 3 different types of bond are formed: hydrogen bond, pi sigma, and pi alkyl bond. Ocular irritation studies showed the safety of lenses to be worn, and in vivo release studies showed that the drug released from the DL direct loaded lenses for 12 hours with improved AUC and min residence time as compared to the eye drops, marketed eye drops. Thus, the study demonstrated that the addition of graphene oxide to contact lenses can control the release of drug as well as improve its lens, swelling and transmittance properties. Thank you.
Thank you, Ditiksha. Would request you to, if you can stop sharing so we can display your video. Perfect. And now over to the judges. And I just have one question and then request judges to ask, how about the safety of graphene oxide? When you have done the studies with increasing concentrations, did you do the safety studies in animals as well to establish the safety margin?
Yes, sir. I have done the ocular irritation studies by following Dre's guidelines, and I had the final formulation which I had taken up for the in vivo release that I have taken, which had maximum graphene oxide, that is 800 nanogram, and it did not show any signs of irritation or redness. Plus, I have also done histopathology, which I had not mentioned here, and there were no signs of any necrosis also.
Okay, all right. So because those slides were not there, but great, you have done that. So okay, so over to the judges. If you have any questions, please.
This is Patanjali. What's the cost of these contact lenses?
Yes, each one pair of contact lens will cost around 300 to 500 rupees. One pair.
And how long it lasts?
Yes, my research showed that the drug will release for 12 hours, but we can modulate this. Still, optimization is required by modulating the graphene oxide level. But if we can give a release for a 2-day or 3-day contact lenses, then we can use one lenses for extended period of time. Now, if we talk about cyclosporine, the eye drop formulation which is available, one bottle costs around it is more than 300 rupees. It is an expensive drug. And even other drugs which are used for glaucoma, bimatoprost and latanoprost. So if we can prepare such a lenses that it releases the drug for even say 7 days, then that will be cost effective and very helpful to the patient.
So Ditiksha, Jignesh here. So
Yes.
good work and novel approach to meet the unmet medical need. So I have a question related with respect to the in vitro release method. So what did you apply to study the in vitro release?
I used a normal orbital shaker, and 2 ml was the media which was used for the study. 2 ml of the simulated tear fluid. Considering the small amount of tear volume which is available in the eye, we had used 2 ml, and we used to replace complete that 2 ml at predetermined intervals.
Okay, and was the in vivo estimation was from tear fluid or blood?
Yeah, no, from the tear fluid.
From the blood?
Tear fluid, sir.
From tear fluid.
Yes,
okay.
Because dry eye syndrome, it is topically, it is on the anterior part of the eye. So we did not collect the blood, we collected the tear fluid.
Okay.
Yes.
All right. So if no further question, then thank you, Ditiksha. And let's move on with the next presentation. After Ditiksha, now we are on poster number 13. Arnab Sinha Roy. He's going to talk about validation of flow cytometry based whole blood natural killer NK cell cytotoxicity assay as immunity marker. So let me quickly invite Arnab.
Good morning, sir.
Yes, I can see you. Great.
Yeah.
So now you should be able to share the screen now. Yes.
Okay. Is it visible, my screen?
Yes, Arnab, you have to do slideshow and
Yeah.
would request you to switch on your video also if possible.
Yeah. Actually, sir, I'm just trying to switch on the video, but there is some issues in the camera. So by the meantime, can I start with the?
Yeah, please. No worries at all. Please proceed with your presentation. Yeah.
Thank you. So good morning, all. Thank you for giving me this opportunity to present this poster here. So I would like to explain that about a method what we have developed and validate in our lab. My name is Arnab Sinha Roy, and today I'm representing Cliantha Research Large Molecule Lab. The method I'm going to explain is regarding one pharmacokinetics biomarker, and to be more specifically, it's an immunity marker, and this is related to the NK cell activity assay. So as you are aware that the natural killer cells are lymphocytes, and these are widely used as kind of a marker to check the immunity status of any individual. And NK cells are is basically actually killing the infected cells and the cancerous cells in a way. The very difference actually it makes like when it's actually attacking the normal cells, but due to the presence of MSC1 molecule, the major histocompatibility complex 1 in the normal human cells, it's not able to target the normal healthy cells, and rather it's actually targeting the infected cells and the malignant cells. So by using this principle actually, so there is one method called the activity assay. So in this assay, we have actually measured the activity of NK cell. It's not the quantity of the NK cells, it's the activity of the cells. So this method is widely used as cytotoxicity assay and primary indicator for the immune function, and there's a lot of applications for the consumer research and the clinical trials. So coming to the method summary, the method what we have developed is has two parts. In the first part is the isolation of affected cells from the whole blood by using the standard fecal-HIPAC method. So we used to go for this PBMC isolation, and that isolated NK cells we are freshly used for this assay. And in the second part of the assay, there is one cryopreserved K562 cells. These are actually leukomic cell lines. And so once we are thawing these cells, we are stimulating the NK cells with interleukin 2. So this interleukin 2 actually stimulating the cells to enable that activity, the cytotoxic effects. And post stimulation, we are mixing the NK cells along with those target cells, which are actually K562 cells. So these target cells, K562 cells, actually lack MHC1 on their surface, which make it susceptible to for this NK cells activity. So these K562 cells are also pre-stained with one green fluorescent dye. And so whenever we are mixing those cells and keeping for incubation for some time, like around 3 hours, and post that we are measuring the dead cells, the amount of dead cells, to check the activity of NK cells by using one DNA staining solution. So this DNA staining solution actually it's actually go and bind with the DNA, and it will detect the dead cells, while the live cells it will be keeping intact with that already pre-stained green fluorescent dye. So we have validated this method. There are a lot of challenges basically during the development we have seen. It's because the immunity of a person actually varies a lot in a kind of a day by day basis, because it can depends on infection and a lot of other parameters. So getting consistent result from an individual is itself very challenging, but still by using quite regulated procedure and very stringent method, actually we are able to achieve high precision and accuracy for this method. So you can see this inter and intra-assay precision data for two individuals over 2 days, and that was performed by 2 analysts. And how it can show actually the inter-assay precision is below 20% for both of the samples. We have also performed specificity, because as a part of the assay, we are using interleukin 2 as a stimulating agent. And we have also used other interleukins to just to check whether there is any possibility of cross stimulation because of other interleukins. And it's very, very specific within certain concentration. We have also optimized the incubation period, and we have validated that part in the robustness experiment. So it is the 5% CO2 where we have performed this incubation, and we have used multiple time duration for the stimulation and also for the K562 and the NK cell mixture incubation. And finally, we have also proved the stability of the isolated effector cells in 2 to 8. So we have seen the isolated effector cells are actually not much stable in -80, and also we have tried to prove the stability in liquid nitrogen, but there are a lot of challenges in there. But we have able to make some kind of stability in 2 to 8. But we can see a lot of degradation over the time for the NK cell activity. So we have validate the method with a freshly isolated NK cells. So this is all about the method validation. We have performed precision, specificity, and stability, and few other experiments. And overall, we have seen the method is very, very precise and stable and repeatable and reproducible over the time. And it can be widely used for as an immunity marker for any kind of clinical trials or maybe the consumer research. So this is all about the method. I'll be happy to answer if you have any query. Thank you.
Thank you, Ashish. So any questions for him? So yeah. I have just one question quickly before judges will ask. You mentioned that the stability was an issue in liquid nitrogen. So whereas you got it at 2 to 8 degrees. So would you like to discuss further on that why on liquid nitrogen? Because generally we see that most of the cells, even from stem cells, everything we do cryopreservation, right? And we do get good stability. So any reason why you did not get stability in liquid nitrogen?
Yeah, so we have actually tried to establish stability in 228 minus 20 minus 80 and liquid nitrogen. So what we have seen, like the higher temperatures somehow the activity of the cells are much lower. It could be the percentage of DMSO what we are using for this cryopreservation. It could be one of the thing, and we have actually used multiple concentration of DMSO for the freezing, but it was not much help. Exact reason even I'm still looking for it. But what we have also tilted is 228 is still much better data, this refrigerator stability in compare with other temperature. And the other part is like it's because of that in cases this isolation also have multiple effect, like how it has been isolated, and in the presence of any hemolyzed cells inside that also kind of interfere with the assay, because we have seen hemolysis has a huge effect, I mean a lot of interference in this assay. So in considering that, I think 228 is the best temperature for now. That's what.
Correct. And while doing the with liquid nitrogen, if there is any way you can get away with DMSO, then I think you may still see some good stability in that as well, if possible.
Yes, sir. Yeah, definitely I'll look into it.
Sure. So over to judges. Any further questions from your side?
Yeah, one question from my side. The method, whether you have adapted from the literature or it's a completely a novel method?
No, sir, it's not a novel method. So it's actually there are few kits available. So they are actually providing the cell lines, which actually are cryopreserved cells we are receiving from one of the commercial vendor. And there are few like BD. They actually provide few kits for like DNA staining solution and the stimulating agent. So it's not a novel method, and it's actually used by. It's actually reported in multiple publications.
Okay, thanks.
All right, thank you. Good job, Ashish. Any other question for him? All right, so in no further question, then would like to thank you, and let's move on to our next presenter. Now we are on, for the judges, we are on poster number 14. And the next poster presenter, the presenting author is Preeti Kumari. She's going to talk about quantitation of monoclonal antibody aggregates in cell culture supernatants by size exclusion chromatography. So over to you, Preeti. You can share your screen.
So is it fine? Is my screen visible to you?
Yes, definitely. And did you, if you have switched on your video, then we can display that as well.
Okay, sir. Is it fine, sir?
Perfect. Yes, please proceed. You're good to go.
Good morning, everyone. This is Preeti Kumari from Institute of Chemical Technology, Mumbai. And topic for my today presentation is quantitation of monoclonal antibody aggregates in cell culture supernatants by size exclusion chromatography. This is part of my MTech project work. Screen is not moving.
I mean, don't worry, just click a couple of times. Sometimes your screen can hang, right? So you will go to the next slide, I guess.
Yes, sir, but it's not moving.
It's not moving. Okay.
Push the down arrow on the keyboard.
Yeah.
Okay.
Thanks, Patanjali. Yeah.
So start with a brief introduction. As we all know, aggregation of monoclonal antibody is a major concern in biopharmaceutical industry, as it may cause immunogenic reactions and it may bring a critical quality attributes. So it may arise at any points during the manufacturing process and at the cell culture stage itself. Downstream processing has capability to remove the aggregates, but if we somehow able to reduce it in upstream, it may lessen the downstream processing. So in this study, we have monitor level of aggregates during cell culture with the help of high resolution size exclusion chromatography technique, and we have monitored the trends in aggregate levels at various stages during the cell culture, which provides opportunity for intervention towards aggregates reduction. We have also demonstrated that the aggregates levels in cell culture can be reduced with the help of osmolytes and formulation excipients, which are generally used for the drug products, finished drug products as a media supplementations. I have used the aggregate detection in supernatant levels, as after the protein purification stage, it may leads to aggregation due to the lower pH value of the due to the lower pH of the elution steps. So materials and method. So for this study, I have chosen the CHO cells which produce IgG, and experiments was done in shake flask. Protective agents like osmolytes and excipients, which may include glucose, mannose, maltose, which is a known sugars and used as a carbon source. Sucrose and polysorbate 80 and polysorbate 20 are frequently used in many commercially available drug products in their formulation excipients. These were added daily from the early exponential phase, and this study was is compared with the control, which does not have any media supplementation of these components. Viable cell density and viability was estimated with the help of automated cell counter using trypan blue dye exclusion test, and samples were collected daily for aggregate analysis using site exclusion chromatography, and titer was also estimated with the help of protein A chromatography. So my main focus is to study the aggregate analysis. So for aggregate analysis, I am using the size exclusion chromatography, and with the help of ultraviolet detections, the analysis was done using 1260 bioinert HPLC system from Agilent technology. The column specification and run conditions are described in these tables. The column specification include the model, which is advanced biosec dimensions, column volume, particle size, and particle pore size. The buffer I am using is 150 millimolar sodium phosphate buffer with pH 7. Isocratic elution was used with a run time of 20 minutes with a flow rate of 0.8 ml per minute. The signal was quantified at wavelength of 220 nanometer, and peaks were determined for aggregates and monomer by calculating the relative peak area percentage. Results and discussions. For each selected osmolytes and formulation excipients, their effects were studied on viable cell density, titer, aggregates, and these all are compared with the controls. So first I discuss their effects on the cell growth. As I discussed, glucose, mannose, and maltose are the readily used sugars, so they did not have any effect on the cell growth, and you can see the growth as comparable to that of the control. With sucrose and polysorbate 20 and 80, sucrose also does not show any inhibitory effect on cell growth. In case of poly 20, you can see a concentration 0.01 shows some little effect on growth at cell concentrations, as cell density is at lower side to that of the control, and in case of poly 80, it is fine and it is comparable with that of the control. In case of aggregates, glucose, mannose, and maltose, with increasing concentration, you can see.
Preeti, yes, you were muted for some reason. Now you can hear you. And I think time is up. So maybe another 30 seconds, you can conclude. Thank you, Preeti.
effects on aggregates, you can see that glucose, mannose, and maltose show the positive effects with the help of aggregates. In case of sucrose, it does not show, as increasing concentrations may lead to increase in the aggregates level. In case of poly 20 and poly 80, its aggregates level decrease with the data of the concentrations increases and the time increases cell culture durations. In case of titer, it's not moving.
Arrow, again the down arrow, can you try that?
Yes, I am trying the down arrow key itself.
Okay, no worries, but I think anyway time is up. So on this slide itself, if you can conclude, no worries, you can just talk about your conclusion. Yeah.
So my conclusion is cytosine chromatography is we can detect the aggregates level on the various culture states. This gives a probability that we can do at upstream production, we can do some requisites to mitigate the aggregates level at the upstream process itself.
All right, thank you, Preeti. So let's go to the judges for any questions. Dr. Shefali, Dr. Amarnath, Dr. Jignesh, Dr. Ravi Shankar.
Yeah, I have one question. Sorry. Yeah, it's a good work. It would certainly help for upstream process. So one of the point here is you are observing very high amount of the aggregates itself. So what would be the reason? Generally, it's like main protein will have higher amount, but here eventually I'm observing about 20% of the aggregates. So when I don't know whether I'm correct or not, but so what would be the reason on that part?
20%. So with the last days of the harvest, that is on the harvest day. So with maybe it may be due to the conditions or like some stress.
But in general, when you go for a production and the upstream process, generally the as such protein will be more, right? So the
Yes, sir,
aggregate
yes, sir.
amount will be very less, but the study showing little bit the other way around. So that's what I was bit curious on that part. Well, why it is so high,
Sir, I think you are talking about the control. In control, I'm just adding the feed and nothing is added. So it may be due to that reason.
Okay.
Okay, so there is one question in the chat. Can SE size exclusion chromatography determine the larger aggregates, or is it suitable only for small aggregates?
It can observe larger aggregates.
No, that's
depending
not true.
on the pore size of the
Correct. That's not true. You have to go for an orthogonal technique, a kind of an AUC or FFF. So yeah, because after certain level, it cannot detect the aggregates, but the other techniques will.
It's other things like AUC and
yeah.
Can also be used for aggregate.
Good work. Yeah.
Sure. Thank you, Preeti. And let's move on to the next presentation. Now we are on, for the judges, we are on poster number 15, Gantashala Subramania Samir Kumar. He's going to talk about the multi-attribute method for biotherapeutics. So let me request Samir, if you can share your presentation, your screen.
Yeah, 1 second.
Yes, Samir, you're good to go. Perfect. Yeah, please.
Yeah. Very good afternoon, one and all. I'm Samir, working as a manager biologics bioanalytical division at USP. And today's my presentation topic is the multi-attribute method for biotherapeutics. Simply and shortly, we can call it as multi-attribute method as MAM. And this is the glance. One can glance this overall view of the poster. And if we quickly go into the introduction, our global biologics strategy at USP is to develop standards and related scientific content. This will be helpful for reference sources to support in adopting the innovative technologies that can further improve the analytical testing, and also it can help in the increase the efficiency of biopharmaceutical development process, and ultimately it can decreases the development cost. If you look at the trends, recent trends and technology, MAM drawn the attention, and it appears that one of the promising approaches that supports the specificity of mass spectrometry to assess the multiple quality attributes at amino acid level. MAM has the greatest advantage in the facilitating unexpected modifications. Also, it can detect when compared with the other traditional methods. If we see the figure 1, these are general selected quality attributes assessed by MAM, and in our study, post-translational modifications results were consistent with various methods across the labs. However, we have seen a wide variation with respect to the percentage of deamidation level, and the possible reason could be sample preparation variation conditions. For that, we have tested different conditions for the reduction and enzymatic digestion steps to see most critical variables that should be optimized to minimize sample preparation induced artifacts. And if you look at this method, this is the typical MAM workflow, includes both monitoring phase as well as the characterization phase. In the present study, we have used and focused completely on the characterization phase using LC-MS approaches to analyze the 3 new USP monoclonal reference antibodies for our further studies. And this is the method where we bulk material can be characterized using various methods across the labs. And here we found that a significant variation was at the deamination level. And the differences in the deamidation level possibly dependent on the sample preparation conditions. So we identified several parameters that could be contribute to the observed variability. And based on this screen, we have screened these parameters based on the literature review of MAM and also various peptide mapping protocols, and discussion with the subject matter experts in the field. And this is the figure 3, where you can see the right side, a 2-phase study was designed to test the impact of reduction as well as alkylation and digestion conditions on the sample prep induced modifications. And this is the results and discussion session. Variability of post-translational modifications. As I mentioned that most of the post-translational modification results were consistent using various methods across the lab. However, deamidation has shown wide variations, and those variations are ranging from undetectable to greater than 40% if you select as a peptide, same peptide, and if you compare with across the labs, and we have seen that much variation. And out of this, you can see in figure 4, pyroglutamate and C-terminal lysine, where it has shown the similar expression pattern in terms of percentage of modifications among all these antibodies. So our focus was drawn the attention towards deamidation because it has a very huge variations were there across the lab. And if you see the impact of pH and temperature during reduction and alkylation, deamidation was shown to increase with increasing temperature and pH. Here in the figure 5, you can observe that deamidation rates were highly peptide specific, because some peptides have shown the high level of deamidation variations, and some peptides were showing the low level of deamidation with respect to the increase of pH as well as the temperature. However, in the pH change as well as the temperature did not show any significant effect on the digestion. We have measured with the coverage as well as missed cleavages and missing of carboxyamido methylations. These conditions we have taken further and gone ahead for the phase 2 study, where different digestion conditions we have opted for our further studies to see the impact of the deamidation. And here the impact of enzyme substrate ratios, trypsin sources, and digestion time we have taken as a primary criteria. And in figure 6, if you see deamidation, this digestion time had the greatest impact, and the digestion incubation time is increasing, and the deamidation rates were also gradually increasing across the maps in different labs. However, we have not seen any significant observations between the enzyme and substrate concentrations and the trypsin source had little impact on the deamidation. All these trypsins used were porcine and were sequencing or higher grade we have used. If you accumulate all these results in a snapshot and higher enzyme substrate concentration and shorter time periods, which minimizes the deamidation during sample preparation conditions. And if you summarize these results, this study identified several key factors that can lead to the sample preparation induced modifications with a special focus on the deamidation. Here we have observed in our present study, lower temperature and pH during reduction and alkylation steps reduces the deamidation, and digestion time had the greatest impact on the deamidation in the digestion step. This method developed in this study will be further evaluated with respect to the results in independent labs across the globe. And this work also provided and laid down the foundation for the development of pre-digested MAB reference standards. Pre-digested MAB reference standards coupled with the existing USP maps will provide the users a great advantage to assess both at the instrumentation level as well as the assay itself for carrying out peptide mapping and MAM other applications. This is my conclusion slide. Evaluation or progress of MAM at USP. Over the past 2 years, this work has been evolved from a single research project to diverse set of activities involved in this. Below are, but not limited to, the support the standard development, stakeholder engagement, and the scientific leadership in the innovative and rapidly evolving field. If you see, look at closely observe the timelines and the activities. We have initiated work on USP maps with external collaborations. We have conducted surveys of various stakeholders to understand the current best practices in the field. We have obtained feedbacks from mass spec peptide working groups across the globes, and of course, with along with other stakeholders on physical reference standards. Around 2020, we have established a stakeholders forum on MAM and additional survey on the best practices. We have recruited and initiated the MAM expert panel. Simultaneously, we have published a few MAM articles, and we have also explored new collaborations to support the standard development. Always, USP stands at the front line to explore or thrive to explore new collaborations, especially fruitful collaborations across the globe, which supports the standard development, which in turn helps in the production of quality products across the globe, which will enhance the promoting the health of the people across the globe. In 2021, we have also replicated and expanded the MAM work in independent laboratory. We have initiated the development of pre-digested MAB standards. Couple of months ago, we have already launched a USP online community of on MAM. Who are interested to know more information on MAM can join and register at mam.usp.org. And this is the reference slide. And thank you. And I'm happy to take up some questions.
Thank you, Samir. And over to judges for any questions for Samir.
Yeah, Samir, Ravi here. So yeah, this is one of the important milestone activities from the USP standard development perspective. So from the presentation perspective, I have a question whether you or your lab has contributed for evaluation of this standard.
Our actually this work has been experimental work has been taken place as a collaborative work in Georgia University in the Department of Complex Carbohydrates Research Center under the leading position of Dr. Azadi. However, most of this data will be scrutinized or reviewed across the global biologics labs of USP. Yes, we are part of that in the reviewing of this data.
Okay. Yeah. So then the second question is, as you rightly mentioned, any variability in the sample processing and the preparation will highly impact on post-translation modification. So what are the control strategies, whether USP is coming out with whatever you have stated as a control strategies to avoid such kind of variabilities?
Yeah, actually, as you mentioned that for any biotherapeutics, especially monoclonal antibodies level, if you take, the stability, structural integrity is very important. And of course, here indirectly the information about post-translational modifications are the key events. And one's supposed to prevent or eliminate the false positives. So what we have done that among all these PTMs in our data, we have found that deamidation has highly variable. So we thought our focus was towards to deamidation, and in the deamidation, especially the reduction and enzymatic digestion conditions, across the labs, we have seen different variations with respect to deamidation. Huge variation was observed. But in fact, if we compare with the other PTMs, deamidation range was around, even some peptides, we have not even detected the peptides with respect to deamidation, and some of the labs has shown 40% variation is there. So this is a critical event, especially with the deamidation point of view. So our focus was on that to see really these steps like trypsin, time periods, as well as temperature, pH. These are all the critical factors about quality attributes. So one's supposed to closely evaluate and monitor to avoid any false positives in the experiments.
Thank you, Samir.
All right. So if no further questions for Samir, then thank you, Samir, and would request you to stop the slide share.
Just to add some little.
Oh, yeah. Sorry, Patanjali, please go ahead. Yeah.
Are you related to famous Ghantasala singer family?
Yes, sir. I'm fortunate and happy to tell that. Thanks a lot for bringing this. Yes, he is my long distance grandfather.
Oh, good to know that. Thank you.
Thank you.
Hope you sing like him too.
Yeah.
So Patanjali, on a lighter note, so is it going to add some marks for Samir?
I don't know. Well, if he sings a song, then it's okay.
Yeah.
Thank you.
And once again, I would like to thank entire APA organization, and especially the Deepak who driven this entire conference in especially tough times, pandemic time, and also the panel judges. Once again, I'm thanks to one and all, and be safe and stay strong. Thank you.
Thank you, Samir. Thanks a lot. So let's move on to the next presentation. And now we are on poster number 16. The presenting author is Shivani Tank. And the topic she's going to talk about is phytochemical rich polyherbal preparation and exploring its efficacy in translational medicine. So let me request Shivani, if you can share your screen.
Sure, sir. Just a second, sir.
Sure.
Yeah. Is it visible, sir?
Yes. And Shivani, just one second. Maybe I'll have to check at my end. Is your video on?
Yes, sir. My
Okay.
video is on.
Yes, we can see you now. So please proceed. Yeah.
Good afternoon to everyone. Yeah, I'm going to present my poster on phytochemical rich polyherbal formulation and exploring its efficacy in translational medicine. I am a PhD scholar and belong to Atmi University. I'm pursuing my PhD in Department of Biotechnology. The entire interest is based on diabetic wound healing. As we all know, the diabetic is a group or aggregation of so many conditions, out of which I've targeted one of such condition, which is wound healing. A simple mosquito bite, if not given proper attention, it can lead to serious amputations, organ amputations or limb amputations, which was quite disturbing, and that motivated me for this Ayurvedic polyherbal formulation preparation. My polyherbal formulation here is likely to show the wound healing property and scar minimization with chronic and non-chronic, non-healing chronic wounds, such as diabetic foot ulcers. The study undertaken would help determination of bioactive phytocompounds, that is definitely based on some polyherbal, rather than focusing on just one plant and one phytochemical. I've taken I've aimed my study for making it synergistic approach by amalgamation of various phytocompounds showing different properties at the same time. The degree and time taken of such wounds is usually delayed, but my formulation is targeted for for complete healing and the healthy tissue granulation, and the time taken could highly be reduced. And the characterization of such novel characters or such phytoactives and its derivatization for further commercialization can also be my chronic wound healing terrain undertaken. The polyherbal formulation is a new concept rather than introducing a single drug or a single phytochemical. Why not to add the synergistic approach to it? I've taken 4 plants under study, which are leucopyrus, that is Selaginella leucopyrus, white X regunda, Azadirachta indica, and Acacia cutichu. Very well known in the Gujarat region, especially very well available under the xeric conditions, and they hold beautiful properties, especially this leucopyrus is very well known for its anti-diabetic property as well. Though my entire concern is now for the wound healing, but the anti-diabetic property would also help patients to keep their glucose level at the wound place very much under control, which would we're soon going to discuss about why there's a problem in wound healing. So there's reduced nitrogen oxide synthesis at the wounds that is actually making it delay the reduced vasodilation. The vessels, the angiogenesis has reduced ability under those cells which are already in tension because of the glucose interference at the wound healing property. In diabetic models also, we have observed that glucose is always
She's frozen, I think. Can you guys hear me?
Yes. One second.
Hello? Am I clearly audible, sir?
Yes, Shivani, we lost you for just a few seconds. You can go back to the last slide and start from there. Yeah.
Okay, so there were various factors. There's not only one factor which is actually responsible for delayed wound healing in diabetes. It is actually multivariant dependent, and some of them I've listed here. There's reduced nitrogen oxide synthesis. There's reduced vasodilation. There are very later or delayed infiltration of macrophages, which are very much essential for blocking the what do you say? For washing out actually the debris which is collected at diabetic wound sites, any wound site to be very precise, and macrophage activation is also comparatively delayed. All of them are somehow directly or indirectly associated with the glucose problem that is being faced in the diabetic conditions. What I have prepared, or what here right now, is the preparation of polyherbal formulation which is going to be oil based, right? And in Ayurveda, this is very much popularly known as telya or til. And this tail could be directly applied as dressing onto the wounds of diabetic patients. So firstly, how it is prepared, the preparation is actually first made with as a quat is the aqueous version of my polyherbal formulation. I have to undergo decoction. There were various hydrophilic and hydrophobic components which I could have been added in of my polyherbal formulation. I've made it in water, as well as I'm going to add sesame oil, which is very much good with antioxidant property and could be very much soothing for the epithelial layer. And I've made my preparation rich in hydrophilic and hydrophobic components. And the pak that I have prepared is mrudu pak. It is actually prepared at a very medium flame, and it is very much ideal for diabetic wound. This is how I've quartz preparation. This is actually Ayurveda mixed with that of plant biotechnology. The dried samples were grinded and made fine powder. Then they were heated under decoction. They were filtered, and filtrate was preserved for a specific period of time. I'm afraid I cannot share exactly the proportion and the time and the process very precisely, as I'm going to apply for patent as well. Then oil was added, and it was allowed to mix for a while. The powdered version, or the raw powdered version, was added 10% of each sample, and the oil was again allowed to reduce to its original volume. And then this oil was filtered, and our polyherbal formulation, our effective polyherbal formulation, was prepared under the doctor Prapti Jivrajani, who is again an MD Ayurveda. Now the formulation was also, these plants were actually given for extractions of phytochemicals. I've taken two polar solvents, water, methanol. I've used this oxidant apparatus, non-polar fractions having chloroform and hexane, and semi-polar fraction petroleum ether and ethanol. This study was targeted to isolate the typical group of polar, non-polar, and semi-polar phytochemicals, and I could narrow down my research to my one or synergistic effect of bioactive component. Further analysis of biochemistry included qualitative and quantitative approaches for phenolics, flavonoids, tannins, alkaloids, terpenoids, steroids, amino acids, anthraquinones, saponins, and glycosides by very authentic methods, out of which we could find that phenolics and alkaloids were of greater quantity, and both of which are recorded for brilliant wound healing properties. I would like to just add glow to my polyherbal formulation. I've carried out my in vitro cell line studies are in the study. There are two essays that I have sent to the University of Miyazaki, Japan, where my co-guide is.
Shivani, sorry to interrupt, but you have 30 seconds to conclude. Yeah.
Yeah,
Thank
yes,
you.
I'm just on conclusion. My guide has carried out two in vitro cell line analysis on hypoglycemic cell lines, in which he made diabetic conditions of 250 mg of normal cell lines, and the cytotoxicity assay on normal as well as diabetic cell line has shows that my polyherbal formulation has passed the toxicity. It hasn't been toxic to any kind of cells. And secondly, macrophage invasion. Whenever scarring was made on cell lines, both normal cell lines and diabetic cell lines, whenever scarring was made, the infiltration of macrophage was terribly increased when my polyherbal formulation was added as a dressing onto those cell lines. So I couldn't incorporate those results here because I just got it in the morning itself, and the photographs were not with me till now, but these were the results and these were the conclusions. So it has been like I'm going to add some more FTIR analysis and read them more, but it is all in progress. I could show this much results for now. Thank you everyone for your kind attention.
Thank you, Shivani. Over to the judges. And I have one question for you, like for wound healing, do you have any plans to check in actual animal studies also, and then checking the efficacy of your formulation?
Yes, sir. I'm going to prepare diabetic mice, and I am going to. We have our animal science laboratory at Atmi University, Department of Pharmacy people handle them. I, for now, do not have any experience on that, on-hand experience, training, but I would take it, and I'm going to do it at Atmi University. The in vitro studies are to be done with Miyazaki University, and here animal studies are going to take place.
Okay, and another question, a quick question is, like, whenever you talk of this polyherbal formulations, to actually pinpoint maybe active ingredients, either one or a group of active ingredients together in a different proportion, that's a big challenge. So did you actually do any of those work with preparative HPLC and collecting those fractions and looking for the structures or something?
Yes, sir. I'm surely going to take it. The major components that I'm targeting for now are phenolics, tannins, and alkaloids, because they proportionally are more in my compound. So I'm going to extract them. I'm going to check their individual wound healing property. I'm going to check, make their mixtures, and I'm going to check for their synergistic approach so that I can know that how are they working. I'm also going to target molecular mechanism of how my polyherbal formulation bioactive compounds are involved in wound healing or macrophage invention or growth factor increment or whatever. In all, if healing is showing, then what factor of diabetic delayed is being targeted, and then I'm going to like how my polyherbal formulation is effective. So what is the molecular mechanism is my next target.
Correct. And analytically, it's going to be really challenging, because I'm sure you would have seen that your single injection, you'll be flooded with at least 50, 60 peaks. So it's a hell lot of work. So so great. I mean, good luck to you. So over to judges. Any further questions? Yeah.
So how did you, this is Patanjali, how did you select those plants? On what basis?
Sir, or with choosing the plants, Kattupilla, or I can say the Leucopyrus was very well. There's a university at Jamnagar which is working under Ayurveda, and I have taken some references from there. I went there with my approach of just interest that diabetic wound healing is my target. They told me there's a there's a plant, Kattupilla, very well known there, and which is having promising. They did clinical trials also, sir. They were doing clinical trials with a single drug of Kattupilla, and then they showed that instead of amputation, like there was a patient who had to go limb amputation, but with the continuous preparation of this Kattupilla dressings and proper analysis and proper care, he revived all of the angiogenesis, and everything was built again. So the tissue regeneration or the tissue healing was brilliant in case of Kattupilla. Secondly, Leucopyrus. What I mean to say is, so Leucopyrus has brilliant wound healing properties, and it also brings diabetes or glucose condition under check. Secondly, the drugs that I have chosen, Vitex nigunda, excellent antioxidant property. Azadirachta indica is for antimicrobial analysis, and Acacia catechu, again, anti-inflammatory property. And this is how I and my guide and the co-guide at Dr. Harish Kumar Madhyastha, we had a panel, and we had taken some expert advices, and we came across that why just targeting single drug, why cannot they synergistically come together and find some. So microbial infections, typical microbial infections of Pseudomonas and Staphylococcus aureus and various other Klebsiella and all, they are very much prominent in case of diabetic wounds, sir. If my formulation, one of my objective is that if my formulation is effective under them, against them, then microbial load can also be reduced, and further infection and pustulations can also be targeted. So that is how we came to this. We narrowed down our research to this 4 drugs, 4 plants. Yes.
Thank you. And Shivani, only suggestion would be that, of course, you'll be doing that, that whenever you plan those studies, always use a control. Maybe allopathic gel or a tube, because there are quite a few many available. And if you establish this polyherbal efficacy against that as a control, I think that will add more value, right?
Done, sir. Thank you.
All right, thank you. Thank you, Shivani. And with that, let's go on to the next presentation. Let me first open. So now we are on the last presentation for today. The presenting author is Abhijit Wadhave. He's going to talk about analytical quality by design methodical approach for quantitative estimation of teriflunomide from human plasma. So let me invite Abhijit. Abhijit, you can share your screen.
Is my screen visible?
Yes, Abhijit, your video is on. I think, okay, yeah, it's on now. All right, great. So yes, it's visible, and only request would be that if you can do the slideshow.
Yes.
Perfect. You're all set. Good to go, Abhijit.
Yes. Good afternoon, everyone. I am Abhijit Wadhave. I have done this research as a film student from Society College of Pharmacy, which is in Mumbai University. The topic of my research was the quantitation of an API in dried matrix spot by validated HPLC method. Going to overview these topics from overall the presentation. So aim of the presentation was to adapt the efficient and convenient method for sample preparation and apply this method in a bioequivalence study. The objective of the research was to develop reverse phase HPLC bioanalytical method for estimation of teriflunomide from DPS, that is dried plasma spot. So plan of work I have done is the literature survey, selection of mobile phase as other chromatographic conditions, optimization of experimental condition for extraction, and calibration studies. So drug profile of the teriflunomide I have mentioned over here. The drug is also called as teriflunomide. Rationale of choosing the drug was that it is a suitable mode because of pure aqueous solubility. It is a limited. It has a limited literature reported and has a good absorptivity, and thereby can be easily quantified by using HPLC-UV as an analytical tool. There was the other rationale of using this drug is to explore this new technique for extraction of bioanalysis. So these are the chart chart of various years which has been articles are published. So triad plasma spots. It is a card. It is a card that is have a 5 spot protein saver or a 4 spot protein saver snap card. This card is ready to already has been to use and provide easy collection, identification, and transport of sample without need for any cost freezer or a dry ice delivery. The specification of the card is there. These are manufactured in accordance with the ISO 9001 with the strict protocols like quality control to ensure homogeneous composition as well as uniformity. So optimization have been done with the solvent I have taken, that is ACN and water, which is in the ratio of 60s to 40, which is having split peak due to the acidic nature of the drug. The buffer mobile phase has been used, that is a phosphate buffer having 6.8 pH, and we got the adequate retention and good peak shape, and having retention time is 3.711. So this is the chromatogram we have got. So methodology of the triad plasma extraction was we have taken 50 microliter pool human plasma and spike it with a 1 microliter of 1000 ppm in a methanol solution of teriflunomide. We have been vortex the solvent for a 30 minute, and then after drying we have added 15,000 microliter of solvent extraction. The mixer is spotted on a protein saver snap part having 903 card and allow it to dry for 2 hours. Then evaporate it to dryness under a steam of nitrogen. Then we have reconstituted with the 250 microliter of mobile phase, and we have injected into the 20 microliter into the HPLC. So these dried plasma spotting and chromatographic analysis of blank plasma have been done, which is chromatogram of blank extract evaluated from a interference at the retention time of teriflunomide and internal standard. So we have tadalafil. We have been using as an international standard, and we have optimized the experiment because of the interference and retention time of drug percentage organic phase was decreased from 80% to 60%. So these are representative chromatogram extracted sample containing drug compared to the internal standard in the mobile phase of comparing ACN is to phosphate buffer and pH 6.8. So optimize has been done is 2:3 full factorial design was used to optimize the extraction condition using DPS extraction technique. So these are the experimental batches we have taken, and finally we have optimized the drug matrix spotting conditions. Volume of plasma are used is 50 microliter, volume of extraction, and the cyclometer has been mixing time has been done with the 30 minute. So calibration we have been done with the 8 solvent we have taken from the stock solutions, and we take the calibration curve for the same, and we got the significant value of R square, that is 978. So conclusion of the experiment was that it has an easy and concise bioanalytical method, and it was been developed for the drug teriflunomide using dried plasma spot as the extraction technique for sample preparation and employing this method in the HPLC as the analytical tool. It has also shown the validation of the development method need to be evaluated as per the US FDA guidance on a bioanalytical method validation as per 2018. And this evaluation of analytical technique suggested that the sensitivity of the HPLC method analysis is very good, and hence the developed method can be effectively used for routine bioanalytical studies. That's it from my side. Thank you for patiently listening.
Abhijit,
Thank
this
you,
is Amarnath.
Abhijit. Over to judges. Yes, Amarnath,
Yeah,
please
yeah.
proceed. Yeah.
So, Abhijit, just a quick question. I mean, any reason why did you not use any
Yes, sir.
isotopic label ISTD instead of using the tadalafil? Are you able to hear me? Hello.
Amarnath, can you
sir,
repeat your question? Yeah.
No, I'm just asking, is there any reason you have used the tadalafil instead of using any isotopic labeled compound?
Sir, instead of tadalafil, there was not the other drug was available in the lab, so I will use that.
And I'm just talking about the deuterated compound, because when I was looking your regression coefficient, it was 0.97. And probably if you use some deuterated compound, the regression coefficient mean further improve to 0.99 even. So that's the reason I just asked this question.
Yeah, yes, sir, yes.
And it's all available commercially.
Yeah, sir. Yeah, sir, I agree, sir. The drug was the part of the academic research, so tadalafil was available in the lab, so I have used that.
No problem. Yeah.
This is Patanjali. There is also kind of evaluation for hematocrit values too for red blood spots. So I'm not sure whether you consider that or not. So some of the dry blood spots which we have developed in our company, hematocrit values are also needed to be considered for the viscosity of the blood and things like that.
Yes, it was evaluated actually.
Somebody can be on mute, please. Whoever it is. Did you hear that from me, Abhijit? What I said?
Hello. So actually, we did not observe any hematocrit effect from the drug. That's
okay.
So Abhijit, means the extended question to the Patanjali's one is that means hematocratic effect it will be observed in blood spot technique actually. It's not in the plasma one. His point is means whether in the plasma did you observe anything like that parameter? Okay. So do you observe anything means lot to lot variation? Did you check with any different lots of human plasma?
No, sir, not in the human plasma.
Okay. So another one is that what are the extraction trials you have taken? Means you told you used 1500 microliter of extraction solvent, right? So 50 microliter of plasma, you incorporated 1500 microliter of the extraction solvent. Okay, it's a huge fold of dilution. It will be there. So yes. Before choosing this extraction solvent, did you went for any exploration? Means different trials you have taken with other solvents?
Sir, for the extraction, yeah, for the extraction we have used ethyl acetate chloroform, but we have found that the extraction was done in the ethyl acetate more compared to the other solvent.
Okay. So did you incorporate any sonication step? Means because in generally dried plasma spots or dried blood spots, the sonication is one of the important steps, which will be keeping for half an hour to 45 minutes. Okay.
Yes, sir. Yeah, we have done the sonication.
Because you did vertexing for 30 minutes, right? Means it would be good if you incorporate sonication step. Means were you aware or means you explored something on that sonication also?
So we have done vertexing and also sonication.
Because that was not captured in the sample preparation, that's why we placed that question. Okay. Anyway, thank you.
Yes,
Abhijit.
we have done
Thanks.
13 minutes.
Nice work.
30 minutes of vertexing.
Okay. All right. So thank you, Abhijit. If no further question, then would like to thank you, Abhijit. And with that, yeah, Patanjali, please proceed.
Yes, yeah. For further, just to help you out here, you should also do spot evaluation and spot volume evaluation too. Otherwise, the method won't be complete for the dry blood spots. So spot size evaluation and also spot volume evaluation needs to be done, as part of the dry blood spots. Okay.
Yes,
So
sir.
if you wanna make
We have. Yes,
Go
sir.
ahead.
Actually, we have that in a process that hasn't done it. So we'll do that next.
Okay. No problem. Thank you.
Yes.
All right. So thank you, Patanjali, and thanks, Abhijit, for your presentation. So with that, we come to the end of this AP India poster session. Let me quickly open up here. Yes. So in total, we had 17 posters and one poster from Zifang Yu from Wuxi Aptech and one from Ashutosh Shaler from Shimadzu. So in total, for the session, for the poster session, there were 15 posters. So judges were to score on this 15 posters. And I think the poster number 2, Ashish Kumar Sonkar, he was not there, so we couldn't really have his poster presented. So in total, 14 posters have been presented today. These are the list of that. And let me quickly show you this important slide also. So APA India poster session, as you all know, we will be having 9 prizes. So we give 3 best, 3 best poster winners first prize. So there'll be 3 first prize, 3 second prizes, and for third prize again 3 best poster winners. So total 9 prizes. So as you can see, there are some cash rewards and a memento and a certificate you get. And this year onwards, for all the attendees, poster presenters, and for the panelists, I'm so glad to share that we have started this new initiative to encourage academia to participate actively in APA India meetings at this international forum. So we have started Professor Saranjit Singh Memor Award for the poster winner from this year onwards. So it will be applicable for this year also. We just kept it as a surprise for all the poster presenters. So apart from the prizes which are mentioned above, this one poster winner from the academia, this is especially for the academia. So researcher from the academia, from out of those 14 poster presenters, one of you would be getting this award based on the scoring from the judges. So you will get a trophy. You get a free registration for AP India 2022. That is next year, and most likely it will happen in-person meeting. So you get a free registration for that, and wherever you are, in whichever part of the country, you get a to and fro economy fare, airfare to fly to that place and attend the meeting. And while you are there at the conference, your 3 nights accommodation would also be taken care there. And apart from that, any local travel which you go from your place to the airport and from airport to the venue, those things will also be covered. So this is an additional award which will be given to the first poster winner selected by the committee. So this year onwards, every year apart from the poster awards, we will also have Professor Saranjit Singh Memor Award also. So this was specifically for academia, researchers from academia to participate and showcase your work at an international forum. Okay, so this is so keep your fingers crossed, and hopefully any of you guys would. I mean, almost 9 prizes are there, so I'm so glad that many of you will get this prize. And before I invite officially all our judges who were there for almost 4 hours with us, I would request all of them to switch on their cameras. And before I request them for the concluding remarks, would like to thank every presenter. You guys have done a great job. And I think all of your presentations were really interesting, and looking forward to your active participation in the next 2 webinars also. But before I make the final announcements for the remaining webinar, let's hear from our judges. So would request each of the judges for their concluding remarks, to start with Ravi Shankar.
Yeah, thanks, Deepak. I think, it was a great presentation from all the participants. And so we have covered in most of the presentations are like we have covered all the areas, I should say, starting from small molecules and towards large molecules. So happy to see many, papers, presentations from large molecules, because that's the need of the hours currently, what we are looking for. And like couple of presentation are extremely good. Like one I can really quote that as a multi methods for upstream evaluation that would really helpful from the application perspective, as well as you can have a multiple application of the various instrumentation which were not in use, I should say. Kind of SACs and other things are recent instrumentation we are availing for characterization purpose for biologics to prove the biosimilarity. So I think all the students have done a great job, and they are moving towards heart area, as well as a couple of really good presentation from the basic purpose. So all the best to all the contenders. So its participation is more important than winning anything, and it's really a good selection and great presentation. Thanks, Deepak.
Thank you, Ravi. Over to you, Dr. Shefali.
Yeah, thank you, actually, Dr. Deepak, for this organizing this very stimulating. It was a very stimulating poster presentation by all the participants, and as Dr. Ravi Shankar shared, that it covered the entire gamut of small to large molecules over so many platforms, and I had a very nice. It was a lot of learning for me as well, and the very good part is that you have given so many, you are giving so many prizes away, so I am sure not people won't be disappointed. The participants, their efforts will be well paid. Some of the posters were actually very good. And thank you so much for
Thank you, Dr. Shefali. Over to you, Jignesh.
Thank you, Deepak. So it was wonderful to be a part of this session and judging the posters, very good posters. We thank all the presenters from academia and industry for being part of this poster presentation of APA India 2021 chapter. And we could see the great work happening in pharmaceutical world, and particularly in academia, and addressing the unmet medical need of the patients. Today's sessions covered almost all type of pharmaceuticals, small molecule, large molecules, and herbal extracts, covering PD, PK, and its analysis. We look forward to all the presenters from academia and industry to be part of the next APA India chapter with their advanced research work. So once again, thank you all the presenters for being part of this session. Thank you.
Thank you. Over to you, Amarnath, Dr. Amarnath. Yeah.
Oh, thank you, Deepak. I mean, first of all, thank you, and to all the participant attendees. To be stay so long today. We started at 8:30 and now we are close to 1 o'clock now. It's almost 5 hours. And first of all, thanks, Pat. I think it's midnight. He's going to maintain the next day. And I mean, it was really a learning day for everyone today. And I'm so happy to see, majority of product is from the pharma industry, and I could see that a lot of the students are working on the same product. So there is a lot of opportunity for the student who are there in the academy to give a path for them to enter into the industry. So I would encourage even the student who are there, and they had a lot of even the great futures ahead. So thank you very much for all the participant and the student who has presented today. Without any agitation. I mean, they were so bold to try to give all the answer. I was also trying to learn a lot of discussion from the Ravi, Jignesh, and all the people. So it was a great session today. Thanks, the Deepak.
Thank you, Amarnath. Over to you, Patanjali.
Oh God, right now it's 2:20.
Oh my goodness.
Interest keeps you. Great. The best way I can describe this was this kaleidoscopic view of all the methods and the science going on in India, and it's an overall view which is phenomenal. I learned a lot today, right from the topic of herbal to the topic of cyclosporin in the eye to the MAMS to the SACs and whatnot, all the methods, So I'm in their outsourcing operations group, no longer working on the bench. This is the only way I can learn things about what's going on in the industry. So my learning stopped with API 5000 or API 4000 now. It's all API 6500, maybe 7,500, something else might come up, right? So, we used to have an instrument which was needed a room of 10 by 15 negative chemical ionization instrument. Now they are all 6 by 5 or 6 by 4 instruments. So miniaturization is the key nowadays, right? So, that's a lot of topics covered. It was stimulating, as Shafali said, and invigorating, science. And I hope my thinking is like, we have here called local state level New Jersey mass spec group or Richmond mass spec group or something. If people like here in this area can have a local, like bi-monthly or once in a 3 months, like a club-like meetings amongst the industry itself, that will help the students as well as the people who are working in the CRO to exchange ideas in the science and technology. Once in 3 months, or like we have that kind of meetings here, where a lot of young scientists come and present and exchange ideas. So you guys can think about forming such clubs, just kind of a thinking, if you guys can put your brains together, like Amarnath or Ravindranath or Jignesh, and even Deepak can help them out too. So where you can formulate, and then from there you bring people to the APA India, and that kind of a growth. And then you involve the colleges there. Right now, Deepak is doing a fabulous job, but he might need your guys' help to kind of start at the basic grassroots levels, wherever the science is really happening in that particular area. So that's my final kind of a, not suggestion, just a kind request. That's all it is. I've been talking to Deepak a lot several times, why don't we knock on certain people, colleges' doors or professors, like, and especially esteemed institutions like Institute of Sciences in Bangalore and things like that. So it's very hard to take for one person to take up this job. For me,
Sure.
Yeah, these are all things. So, and good job, Deepak. And then the Russ's talk was really fascinating. So that's my second life. It's going to be an NGO. If I retire, I'm going to be part of that kind of organization to help people out. And I hope you. Yeah, I hope some of you might know I'm also a patient with a kidney transplant done in 2017, surviving for 4 years. So, and cyclosporin is my drug. Every day I take that drug, 75 in the morning, 75 in the night. So you guys are keeping me alive, with these kinds of drugs. So I'm not only a scientist but also a patient, being served people like you. So that's what motivates me to participate in this APA India and help as much as I can. And I also mentor certain APA India scientists who have gone on to different roles in different companies. So you guys can also do that. Thank you very much. Have a nice day. And sorry, India couldn't win the 2020 tournament. I was so disappointed.
No, I think the entire country was equally happy when Australia win the semi-finals.
Oh. So that's sad. That's sad. That is sad.
I think Pakistan really played well this time, and
no
doubt about that. I mean, they were great.
No, they needed that victory more than any other country. You know why? That country needs some kind of a. That country is not progressing well, so this might boost them in economy sense or something. That's what my thinking was. I know I don't want our rivals to win, but in a different direction. If you think that way, is the neighbor, right? So if that country wins, there are a lot of opportunities for the people around that area. So Australia is common, New Zealand is common, right? They don't need any boost or something like that. But Pakistan winning a tournament might bring a lot of money for that country. So that's
I agree. So at the outset, let me just now thank Patanjali, especially on behalf of the committee members also. Like 2:30 a.m. in the morning is a big deal, Patanjali. We really appreciate and your contribution to APA India. I think you are there since the inception of the APA India.
Yes,
so on behalf of the entire committee, a big round of applause for you, and thank you for your support.
You all too.
Really appreciate. And now, thank you. And let me now thank all the panelists, all the judges for staying so long, for 5 hours at a stretch. Thank you, I know all of you are so busy, but still you could spare some time for this students, researchers, and posters from the industry. So thank you all the panelists, all the judges. And one last thing, let me request everyone, all the poster presenters, if possible for you, can you just switch on your videos so we can just take one snapshot and then we can finally sign off. So if all of you can just switch on your video, we will try to take one snapshot. I don't know how many will come in this gallery, but yeah, quite a few many I can see. All right. So we have Rahul, Preeti, Ishwara, and of course, before I go to the other thing, how can I forget? We are equally thankful to Shrikant, Dr. Shrikant from Laksai for being one of the judges. And although he was traveling, but he made it a point that he attends. So before I take a snapshot and others are just switching on their video, can we have concluding remark from Shrikant as well? Shrikant, over to you.
Thanks, Deepak. Everybody has conveyed what it has been happened in the last 3-4 hours. Learning is a journey, and we are all on this in the process of that one. So all the sessions it's quite engaging and enlightening. That is the minimum and brief remarks what I can give. Thank you all. Thanks a lot.
Thank you, Shrikant. Thanks a lot, and really appreciate. So I think Shivani, very quickly, we are glad to have Dr. Vijay also with us from Nectar. If you can switch on your video, Anita, Ditiksha, Anita, you can also please, your student has presented today. If you can switch on your video. Vinod is also there for our committee member. He can also switch on. Shivani, Shikha, and Zifang. I know it is too late. If you are there, please switch on the video. And Shikha and Ditiksha, if you are there, can you just quickly switch on your videos? Okay, so I can't complain because it's been a very long session. So I can understand if others are not there. So let me just let's say cheese and let me take one snapshot. All right. So thank you, everyone. Thanks a lot for being such a sportive, active participants. I'm in great session today. In advance, congratulations to all the poster presenters. You did a great job. All of you have presented well. And thank you to Jay. I'm seeing him after a few many days. So thank you, Jay, for being there as one of the organizing member. And Vijay sir, Rena sir, I think we need more presence of yours in coming webinars. So with that, I think let me just conclude and sign off here, and wishing you all a great weekend. The results for the final winner, we are going to announce in the last, which is the 27th November webinar. But would request all the poster presenters to please attend next week webinar also. We have got a very interesting talks lined up on next Saturday, which is 20th of November. And then we have our the final concluding webinar of APA India 2021 conference series. It was a webinar, total 18 webinars were there. And out of that 18 webinars, I think today we have done 16th one. So 2 more to go. So a very, very long journey of almost 6 months. We started in June and now we're going to end in November end. So would request all of you to stay connected, and we will be announcing the prizes on next Saturday or the Saturday after that. And today we did not do a Kindle draw because it's too late. So I don't want to hold you guys anymore. We will do this. We will do 2 additional Kindle draw in the next webinar, which is 20th of November. So instead of 2 draw, we'll do 4 draws in the next webinar on 20th of November. So please join that. And once again, thank you all of you. Have a great day and God bless you all. Thank you.
Thank you. Thank you, everyone.
Thank you.